have 12 hours sharp, so I think we can start. Please do to be welcomed here in Olomouc at the 9th Advanced In Silico Drug Design Workshop. We ran this workshop together with my colleague Pavel Poliszczuk, who will be doing quite a lot of tutorials, and he will have the challenge at the end. But here I've listed all the people who helped, or you will see during the workshop. So there is quite a lot of effort going on. So I hope that you will enjoy it and enjoy this week. We provide this as a free of charge, and we can do that only because of the sponsors, where the majority is sponsored by the Elixir Czech Republic infrastructure, but also we have support from the EATRIS and CZ Open Screen infrastructures, and from the Aurora Alliance and through the Erasmus+. So, and of course, I have to specify that we have a support and sponsorship from the Rowan, who will be not only having the first lecture, one lecture today, but they will also have a workshop tomorrow, and they provided us with the challenge prizes. So, you are here in Olomouc at Palacký University, which is the oldest university in this part of the world. And it was established 1573. And I hope that you will enjoy not only the university, but also the city, because according to the Lonely Planet Guide, we are the secret jewel of the Central Europe. So here are the important points. We are here at the Faculty of Science, and during the time we will have the excursion here at the IMTM, at the Faculty of the Medicine. And of course, just to orient you, here is the train station. Since we are providing with the coffee breaks, but not with the lunches. Yeah? So you can find several options where to go to lunch, and the closest one is Bistro B6, which is in the 6th floor. Or there is a buffet downstairs in the 1st floor. So those are the closest options, but there are several cafeterias and several restaurants in walking distance, because this is a student city, so the lunches are relatively cheap. So, according to the program, today we will be only here and we will have the lectures, basically about the introductions. And we will have in the evening a poster flash talks. So you can still put your poster flash talk into the document. You can find it on a Discord server or on the webpage that there is a link to the Google Slides. And you can add there your slide talk, which will be one slide and 3 minutes. And afterwards, we will have the poster session with some snacks. Tomorrow, we will move on to mainly onto the structure-based drug design, and we will move towards the alpha foldology and quantum mechanics and machine learning. And there will be 2 tutorial sessions parallel. And we were thinking about how to deal with that, how to set up. And we basically decided that the easiest way will be first come, first serve. So one tutorial on the computers, which is on the orange color, and that will be the Pharmacophores by Thierry Langer, will be just next to this Aula 3002, which is our computer room. And we have approximately 30 computers there. But you can, of course, install and work, and there are approximately 40 places. So you don't need to use our computers, you can use yours. And there are the installation instructions already on the web page. And the second one will be across the hall, and that will be the online tools. So you don't need anything to run them, basically, apart of your computer. And that will be the Rowan and also the online structural analysis with Prankweb, Mole, and Vena. Then in the evening, we will have a conference dinner in Kozlovna, but the capacity is unfortunately limited to 80 people, and we have over 100 registered on site. I don't see that many people here, but that's up to yet, so maybe they will arrive. So if you haven't registered for the dinner, there might be still a possibility that you will fit, but we'll see what we can do. On Wednesday, the aula is used for a different conference, so we will move to a different, to the 5th floor, and there is a room similarly big, but not that nice. And there we will have the lectures about mainly in a field of docking and this structural drug design. And there will be 2 tutorials, and those tutorials will be again in rooms 302 and 305. On Thursday, we will switch to the ligand-based drug design methods into QSAR and de novo design and chemical space. And this will be really short session for the lectures, and most of the time we will have handout, but in this time it will be these 2 rooms. So instead of the 305, it will be the room in the 5th floor. And on Friday, there will be a challenge, and that's up to Pavel. And that will be basically a selection of the active compounds from the pool. And he will give you the information. The informations are already on the webpage, but the majority is that it will be the selection of 100 the most actives out of the list of approximately 3,500, something like that, for a for a target. Even I don't know which one it is. Hopefully, I hope you know. Oh, that's slightly helping. Okay, do you have any question to the program?
Regarding the second session doesn't mean that it will be always online. So in some sessions you will need to install some tools, so please check the Discord channels and websites, and we will provide details how and what you should install. Just an important note, so it doesn't mean that everything will run purely online. Regarding the challenge, yeah, as Carl told, you have 4 days before Friday to create some small teams, up to 3 people, to participate in the challenge. So it may be easier for you to compete with others, but it's not necessary. So you can try to do this alone, this challenge. So we provide some prizes from RowanSci for the first 3 teams. And this challenge will be also available for online, but the leaderboards will be separate, on-site and online people. We will separate. So that's all from my side, maybe, what I wanted to add. I just want to wish you enjoy this workshop, find new friends, colleagues, maybe establish future collaborations here, and just enjoy the city. Thanks.
Okay. So as I've told, all the materials we collect on the webpage, but also this year we have a Discord server. So this is a QR code which leads into the invitation to the QR server. So if you aren't there already, it's really helpful because it's the quickest communication platform. Yeah. Because otherwise our emails with Pavel will be completely filled, and they are already. In addition to this, you see that we are now streaming live on a YouTube channel, and the YouTube channel has its own 9ADD playlist. So all of the videos which will be either streamed as the tutorials, as the lectures are, there will be either streamed directly to that playlist, or we will upload them later if the session will just use Teams or the Zoom and without the streaming. So we will upload additional recordings to that playlist as well from the other tutorial sessions. So just to try, is there a question on me on Discord? A general question. When they will start? We started. Okay, and that's nothing else came up. So, with that, do you have any questions, further questions? If not, then enjoy the workshop. Okay, so, and in the course program, there was a lecture done by me about the drug design introduction. So let's switch to that. I really like the short book by David C. Young about computational drug design, which is approximately 2006, I think, or 2009, which is completely outdated these days, but it's still quite nice, nice introduction into the concepts of the drug design and why the drug design by just puring the AI doesn't really, will not be really much more quicker. And we have to think about the in silico tools as a tool. Similarly, as the organic chemist is using NMR machine to get knowledge about the structure and characterization. So you can do the characterization without the NMR, like using the mass spec, for instance. But if you have the NMR, the loop is usually much quicker and it's much better to use those tools. And if those tools are already here, it can help, but you have to know how to work with them. And that's basically the part of the David C. Young motto. So in this lecture, I will be speaking shortly about where we get the drugs nowadays. And then I will switch to why this designing of the drugs so expensive and hard. And then I will move towards the drug targets and how changed those drug design strategies can be. So last year when I was trying to use the AI to generate the infographics, it was not working. This year it is. So just puring the text into a notebook LM, I was able to generate this nice graphics, which I think sums up quite well the field. And that's at the beginning, those medicines were basically natural observations and traditions. Then in the 18th and 19th century, the rise of synthetic chemistry allows us to get the pure substances that can be used as drugs. And then during the 20th century, we started to get, in 50s and 60s, there was a huge boom of new synthetic drugs, and somehow they found out that it's not really always beneficial to have a new drug, especially when when usually thalidomide affair is taken at the time, when instead of using some kind of a medication to help the pregnant women, you generate the children which don't have the hands, because the compound was actually teratogenic, and therefore the administration and checking of the drugs started to be much stronger and much more pervasive. And currently, we are trying to understand it with the digital biology and with the specific personalized medicines, and trying to get into the details of how the drugs are actually molecularly working, and trying to find this way how to specify the things up, which of course makes the development of new drugs more costly because of the regulations and also because you have so much things to test for. Yeah. So the screening really is, in most cases in the past, was that only 1 out of the 10 of the compound which started the clinical trial actually ended up well. And this one started to be shortened up by the more successful preclinical and more successful clinical trials, because since we know more about what to test for, we can do that pre-front. So for instance, during the years 2000 and to 2010, the largest problem was the drug metabolism of the compound, which basically makes the compounds fail because they came up from the clinical trials as more toxic. Now, the drug metabolism studies are done immediately when you set up the compound itself. So you basically push those things you know that will come up later. You are trying to push them in parallel to the lead optimization to find out the drug as soon as possible. And with the omics, where we have now not only proteomics and genomics and transcriptomics and whatever other metabolomics, of course, lipidomics. There is so many flavors of what your drugs can do and what the disease can do and have, that trying to understand the markers to basically know which drugs you can put somewhere, that's a nice and good challenge for any new big data approach for the drug design, I would say. So as the vocabulary came up, the target, I was speaking 5 minutes already, is basically the biomolecule which the drug compound is interacting. The lead is a compound which we've selected that we will try to make the drug out of it, and we basically optimize that one. And we try to optimize that one usually by using some kind of screens, and from those screens we get the hits, which are the compounds which came up against some kind of a threshold, and we basically try to find out a compound by selection against multiple different criteria. And they will become the candidates, and for the candidates we do either efficacy or the activity analysis, and that's one of them is qualitative, so it is a hit or not, and the activity is usually measured in the IC50 or EC50, which are the effective concentration when the drug binds at 50%. And usually the organic chemists and medicinal chemists are trying to get as low as possible with the activities. Because, for instance, if you take the paracetamol, Paralen, acetaminophen, there are all three names of the same thing, the typical dosage you get in one tablet is 500 milligrams, which means that, and usually that one has an antipyrotic effect. But if you want to have it active against also to help you out of the pain, let's say, then sometimes it's prescribed as 2 tablets at once, but not more because it has relatively low hepatotoxicity. And it means that you take 1 gram of the drug to be effective. And that's because the paracetamol has the activity in hundreds of micromolar effectivity. So if you want to have smaller amount of the compound to produce production and for the efficacy, you need to have smaller amount also in the activity. So you have, ideally, you would like to have something in picomolar or nanomolar range. And it's not really easy to get there with the small molecules. And also, the molecule has to be able to get to the target, so it has to be bioavailable. So the typical drug approval timeline pre-COVID era was this. First, you had the preclinical trials in lab on animals, and then you had the first phase to set up the safety. Then the second phase with the hundreds of participants to get safety and immunogenicity, and then the last one to establish the efficacy on thousands. Of course, that took multiple years to reach the goal. And the problem is that your patent started to get active here. So as quickly as possible you reach to the goal for the approval, you will have more time to actually get something out of the new drug on the market. So longer selection, longer troubles with those other phases leads to much shorter time period for the actual getting something back to pay out for the R&D. That started to be a little bit accelerated during the COVID era, because if you start with the pandemic, which affects millions and hundreds of millions, then it's much easier how to get the participants to get. And they've also accelerated the stuff in the sense that you can combine those individual phases. Okay? So you basically start the phase 1, and after a few times, you basically, if you have a first information about the safety, then you basically start to go to the phase 2, and then you start to go to the phase 3. And by that, you basically are able to reach the final goal earlier. And this accelerated drug approval times means that it's useful when you have pandemic, of course, as we had, or if there is if there is some kind of a disease which doesn't have any effective cures as well. So in that case, you can go to those breakthroughs and there more quickly, because longer selection process will mean that those people who have this incurable disease will suffer for longer. And we don't want them to suffer. Okay? That's the basis of the medicine's things. We want to help people. When I've looked into the data about the new molecular entities approvals by either FDA in the US or European Medicinal Agency, you can see that around 2010, the approvals were basically something like 30. Then those accelerated approvals started to take care, and now we have, the last year, it was 46 new medicines. And in case of the European Union and EMA, it was 38. So we have slightly smaller amount of the drugs which came there. But actually, quite a lot of those things, quite a lot of those compounds came up as a first-in-class. Those are expedited. And this can be expedited especially if you have some orphan disease. Because for orphan disease, which is the disease which is for the rare diseases, then you don't need to do all of that testing with hundreds and thousands of participants, because sometimes on those diseases, those who are really affected by that specific disease can be hundreds per European Union. Yeah, so of course, you need much smaller groups, because you want to help them earlier and basically to have at least something for them. The expedited, of course, was usually around 2001. That was 70% of the drugs were expedited in that year. Of course, again, because of COVID, now the expedited is approximately 40 to 30%. And it comes up to the well-known Arum's law. You probably are familiar with the Moore's law, which is used in computers. That means that once per 2 years, you basically get twice as much of transistors on a chip or something like that. In pharma, there is Arum law, which means that for the same amount of money, you get less drugs in 2 years. And as you can see that this trend actually started in 50s until 2010, it was almost without any change. There was small fluctuations, but the trend was quite solid, I would say. And there were multiple problems, like the pharma companies had the problem with the better than the Beatles, which means that once there was already a drug, then other companies were trying to develop a new drug for that field, which was more expensive for them, and they did get less ROI, return on investment later. Or then there was the cautious regulator problem with the thalidomide and such. So it was tendency to basically put there more money to speed up the things, to have more screens and stuff, and basically use the brute force. It was not working. Finally, around 2010, if you add additional points after 2010, it seems that we reached the minima, and the situation is not really improving, that we would go back to the 50s with the payments, but it seems that something happened. And what happened is actually the usage of the in silico. And usage of in silico and basically trying to find not only in a preclinical way, but also during the phase 2A trials. So you basically, the in silico medicine was able to reach to the phase 2A earlier than previous stage, because they basically assigned the machine learning onto the clinics phase, not only to the preclinics. And of course, the clinics is much more expensive than the preclinical stage. So helping there as well. So it seems that the industry is actually getting slightly better with the return on investment from 4% to 6%. But it's only because of those drugs, GLP-1s. So those obesity drugs are actually distorting the market in the sense that they were a huge success, and unfortunately not the rest of the field. It's basically where it was. So we still have to work a little bit more to get through this. So the question about drug design problem started with this Emil Fischer, Nobel laureate, who was thinking about the drug designing as to setting a silver bullet, which was, in his idea, it was a molecule that would bind to the protein, and this binding would basically solve the disease. Nice idea. Unfortunately, the expensive one. The identification of new drug is quite expensive, as we've seen with the Moore law. And there are also, with the more new drugs are usually more complicated compounds, and those ones are more hard to actually produce. So not only on the development, but also on the production part, we have the higher expenses. And it's also a hard problem, because to find out the drug target pair is not simple. And we now know that we have to take care about the ADMET. which is the absorption, distribution, metabolism, and excretion and toxicity, and side effects coming out of that. There are multiple different obstacles for different drugs development, like if you have a human disease, you don't have any other test cases than humans. So, for instance, there were multiple trials how to get to the humanized mice that could be used for testing of the drugs against the human immunodeficiency virus. None of that actually succeed. So finally, now we have organoids, which are something slightly bigger than just the cells, because we had cell types and cell analysis, but now we have a organoid test where we have small organs where we can test those drugs on screening way. It's new sci-fi in this case. There are multiple rare diseases where the future sales in the previous scenario wouldn't be possible to basically pay off for the cost of the R&D. So thanks to this, we actually have those orphan drugs now, because that was taken by the regulator, and now those drugs are able to go through this layer to find out some orphan drug and push it via the orphan regulation. And only afterwards, if the drug starts to be is registered, then it's possible to basically enlarge the possibilities that it can be added, the drug actually can be found out as a repurposing, that it actually can work against this disease or this disease or this disease. So it can enlarge afterwards. So especially smaller companies are moving towards the orphanage, because the orphan drugs can be developed by middle-sized company. And then it can be enlarged in what it can be used for. Since we are looking for the clinical trials, in all cases, clinical trial 1, 2, and 3, there was the word safety. So we are trying to be as safe as possible, which means that we end in 2 toxic compounds with bioavailability at the end. Okay. So we basically throw all of those which might be active as to be too toxic, and we throw them away. So, for instance, this acetaminophen, the paracetamol, wouldn't be possible to get his registration today, because he would be taken as too hepatotoxic. So we wouldn't have it. And already the active compounds can be already patented. So there was quite a nice way how the weird pharma companies were trying to get over it, and that was by adding of the nucleotides into them. So instead of hydrogen, to put there deuterium or carbon C13, and then they were trying to say that this is a new drug, but of course with the same mechanism of action, while overcoming the patent obstacles, but I think that hole started to close. And of course, depending on the illness type, you will have quite different modes of actions for those different drugs to be tested. The most typical thing which we do is that we do the inhibition. So for instance, you have some cancer type, some kinase is active a lot. So in that case, you are basically trying to find out the inhibitor. And that's easy because, well, it's easy. It's sensible in a sense that, okay, you have a kinase and you basically set up the molecule which will bind the strongest to close and basically block this kinase. So there is a huge development in kinase inhibitors for that reason. Another possibility is you have a high response for the receptor and you basically want to block it. So you are developing the antagonist. That's basically the similar idea. Basically set up the molecule which will bind strongly more strongly than the mediator molecule, and you basically close that thing. But what happens if you have a mutation that basically spoils that protein, spoils that axon, and you basically want to increase the activity of that compound, of that protein? Like you have some kind of a channel which is mutated and works half-time. So to set up the molecule which will be agonistic, that will help this channel to work on 100 capacity, it's much harder task. And that was the reason why the sickle cell proteins were actually cured only with the CRISPR, the first CRISPR therapy, because they were able to use the CRISPR on the blood outside of the body and return the red cells back to the blood with repaired mutations by CRISPR. Yeah. So that was the first CRISPR therapy. I think it has 2 years already. But the problem is that you need to get the CRISPR somehow into the system, and putting the CRISPR inside your own bodies does not work that well. So you have to take it out, work it outside of the body, and then return back. And that's definitely not that easy therapy for everything. For blood, it can help. Then you can have the regulation peptides, like the CGRP peptide for curing the migraine, or GLP-1 analogs for obesity and for diabetes. Those are peptides, and the problem with the peptides is that you cannot take them usually as a tablet, which is the typical way how you get the drugs. You have to get the active form of the peptide into your bloodstream, and that requires you to basically inject those drugs, and that's nothing you might be doing that well on your own. So there are several ways how this can be done, by subcutaneous putting, or you go for the hospital to get the drug there from the infusion. But that's everything like this will not scale, because you cannot put this on the whole population easily, because you need the medical assistant, you need the nurses, don't shoot them, for instance, and basically put those drugs into your body. And of course, you don't need to speak specifically only about the proteins, but also to the other biomolecules like RNAs. And using the RNA inhibition or RNA aptamers, that's a new field, and we've seen with the mRNA vaccines that the RNA therapies are slowly getting the traction. But mainly in this workshop, mainly we basically speak here about small molecules with proteins, but not only. So what are actually the drug targets? If you look into the drug targets for the clinically approved compounds, you basically find out something like this. So it can be done on Campbell, for instance. And when I did this analysis in 2016, we found out that approximately quarter are membrane receptors, then approximately fifth is 15% is the ion channel, 10% transcription factor, some other enzymes, some specifically to the DNA, and so on and so on and so on. So how much of those targets are there? In 2016, Ron Santos and John Overington made a comprehensive map where they've tried to find out what drugs are actually used for the individual diseases and what are the targets. And they were able to find that the most typical target is actually GPCR. which is 7 transmembrane helices, active site on top, and once the molecule binds there, it transforms the signal inside to the cell, and that the secondary signalization basically starts some process. You can see that the compounds which are developed against this type of targets was in 2016 smaller than the percentage of the drugs which were using it. While, for instance, the kinases, I've said that the kinases are a simple target, and there was a larger amount of compounds which were tested as a drug than to the actual drugs. Okay? So sometimes, and of course the proteases, that's because of the HIV to some extent. Okay? Because there was a huge effort to get the protease inhibitors for the HIV. The development of those individual classes was also quite different in time. So for the GPCRs, it seems relatively stable, but for the nucleic receptors, it basically slows down in the newer era. And for the kinases, it started to accelerate. So it was basically the analysis at that time. Now, the authors basically moved into they tried to pursue it further, and now they put it into the integrated resource to understudy the human proteome to basically show up which human proteins are known as targets, either clinically, so those are the targets with approved drugs with known mechanisms, or chemically, that there are some potent small binders, or biologically, that we know about their biological function, and we have approximately 30% of our proteome unexplored dark proteins for which we don't know much. And you can see that we have something like 20,000 proteins, and and out of those 20,000 proteins which we have, only 700 are currently known as a target of some drug. So there is a huge amount of the proteins which we can try to drug, let's say. And and the good thing is that we also have new modalities how to attack those previously undruggable compounds, and that's basically by specifying to attacking protein-protein interactions. So we are not attacking just one protein, but we are trying to block their interactions, like the protein p53, which is quite often known as a protein in quite a lot of cancers, or basically trying to go into the RNA. So basically stop the expression of the protein in problem. So the largest difference between small molecule and the large molecule is, of course, their size. But this size has multiple different things. The first one, if you have something as small as aspirin, then it's relatively simple to synthesize this one. Yeah. So it's relatively simple for the production afterwards, and the production of the chemicals on that level, we can have quite a huge and good manufacturing practice, and we can get a nice purity and everything. While you set up the monoclonal antibody like this, it's much bigger, and we are producing them usually in some kind of biological system. So the monoclonal antibodies are produced usually in an organism to produce those monoclonal antibodies, and we are just taking them out. And it's much harder, or at least it used to be much harder to basically get to them. But as I will show tomorrow and during the alpha foldology, to set up a new protein, to design a new protein against any target, has, at least through the AI, started to be incredibly quicker and quite, I would say, almost easy. So if we look into the what type of the compounds are actually used for the as a new drugs, we can see that the majority of the compounds is still small molecules. In both years, in 2023 or 2022, in both years, it was more of the compounds were actually small molecules. But there were also quite a lot of proteins in both cases, and there also started to be oligonucleotides as well. So those type of the drugs starts to be produced, and they only starts to grow. And because of the their selection has now been quite improved with the AI tools, I suppose that the movement in this modality will increase in the coming years. So I'm coming to the take-home message, and that's that the drugs comes from various sources, and they are quite hard and expensive to generate, and mainly because the biology of the humans is hard, and also because the clinical trials are quite costly. We have specific groups where we have the most typical targets, like those GPCRs, ion channels, nuclear receptors, and kinases. But there is a long tail of possibilities of other proteins, which are those orphans, and they start to be more and more important for the drug design. And biologicals are much more complex than the small molecules, and therefore it's much harder to actually produce them. And the final word, there is no one single gold path that would work on every problem, but depending on what the problem actually is, you have to select a modality that will actually work on that specific disease. And with that, thank you for your attention. And if you have any questions, then Indra is here. Or on Discord, I see no questions still. So is there some questions from the audience? Pavla, do you have questions? Okay.
Thank you for the presentation first, and I have more like terminological question. In case you also developed delivery mechanism, is delivery mechanism considered part of the drug, or are they developed mostly separately, and do they have same approval process as drugs themselves, or are they or are those processes different?
Drug delivery options are usually also, they have to be also approved. And the drug delivery itself is an additional field, basically, because quite often you have a drug, for instance, for those GLP-1s. Those are peptides, which are analogs of GLP peptide, glucagon peptide, which means that if you would go through the classical way, through tablet to go into the stomach, then in stomach they will be destroyed by proteases. So it's impossible to use the common way, and those drugs, this is typically like Ozempic, they have to be used, basically use the injection to stomach, do břicha, to the belly, to get the drug there, basically. which is, of course, not that nice to do, but if you want to get loose, or if it's because of your diabetes, then of course you need to do that. And the companies are quite trying to came up with the tablet for those ones, basically by mixing it with some kind of the protease inhibitors, or to enclose them inside the lipids, lipid particles that will be closed behind, and therefore the protease will not be able to reach that, and the lipidic particle would be able to go further, or to use some kind of spray that would go through the lungs. There are plenty of different modes how you can deliver the drug, and those ones are checked as well, but that's a separate, I would say, drug delivery design session than the drug itself. Because the drug, typically you try to find out whether it will work against your target.
So in practice, development of delivery system like liposomes or something like that is separated from drug design itself, from the design of the drug it should deliver.
I wouldn't say it's completely separate. I would say that it's another layer of the drug design itself. Okay. That in some cases it has to be done as well during some time to reach the goal. Still no questions?
Yes, I have one question. You mentioned that paracetamol would be nowadays considered too toxic for production. Yes. How come it is still produced then? Or how does it work?
Well, if you would take not twice the amount of the drug, but I think the limit is 5 times more, then you are killing your livers. So there is a relatively tiny window before it will start to be hepatotoxic itself. Because during the metabolism, the acetaminophen is generating via some cytochrome P450s the NAPQI, which is toxic for the livers. And it's generated in the livers from the acetaminophen. So especially if you are drinking a day earlier, then you increase the levels of that protein, and then the hepatotoxicity, the dosage is lowered because you are actually actively making the hepatotoxicity out of the drug. So
very
don't drink alcohol and then use the paracetamol to cure the headache. Yeah, that's the most important part from this lecture, I would say.
Thank you very much.
Okay. There are no further questions, even in the... Oh. No, there's a general question, not nothing to me. So we'll switch for another presenter, and that will be the Professor Johannes Kirchmair from Vienna University.
Yeah, thanks so much, Carol and Pavel, for inviting me to tell you a little bit about chemoinformatics or chemoinformatics. So just a little bit of a background. I tried to have one slide that just summarizes what we do in the group. So I generated a word cloud from the PubChem or PubMed abstract of our group. So as you see, we do a lot of different things. We basically develop and employ computational approaches, whichever computational approaches it takes to address all the challenges that Karl just pointed out so nicely in drug discovery, early drug discovery. So nowadays, it happens that my group is drifting towards machine learning because the composition of the group is going in this direction at the moment. But nevertheless, all the other parts that are not so machine learning heavy are also particularly interesting still to me is actually my background is pharmacy, so I come very much from the applied drug discovery point of view. This is a more structured point of view on the adventures that we do in our group. So we develop and employ these computation methods to come up with interesting compounds using virtual screening approaches. And then we also employ de novo design, protein design, whatever it takes to come up with interesting compounds. And if you have listened closely to Karel's presentation before, this is where the fun starts, because once you have compounds, you're running basically through a maze of obstacles on how to become to get compounds that match all your properties, in particular also pharmacokinetic properties and toxicological properties. So, as Karel pointed out quite nicely, nowadays it's still very challenging to find new drugs. It's like finding the perfect partner. It takes more than just somebody that has a lot of money to make a good match. So potency is maybe the most obvious thing that we need. We start from high potency, but then there's lots of obstacles, because as we increase potency, for example, it's very likely that solubility decreases. This is an observation that is often referred to as molecular obesity. So this is also something that is existing in molecular space. As we proceed to optimize the potency, usually we get the solubility problem, and with this perhaps an absorption problem, and so on. So what the computational approaches try to achieve is not just getting us through this maze. That said, we don't even know if that sweet spot here that we need for a drug is actually existent, right? This is very different from engineering, from car industry. If you put a lot of money into developing a new car, you will end up a new car. Hopefully it does not look like some monster drugs or what they have recently tried to introduce to the market, but you will end up with a truck, right? But not necessarily with a drug. And this is where artificial intelligence and computational approaches come in. They try to de-risk the process. And one thing that we might oversee a bit is this is all an issue of also being efficient in a way that we need to come up with the new drug swiftly, because the patent protection only reaches so far, 20 years usually, and after that, the value, the economical value of the drug for the producing company reduces drastically. So we need to maneuver this chemical space not just successfully, but also swiftly to gain revenues to push further research. Now the problem here is that the chemical space, and here is actually one of the experts sitting, so Pavel has looked into this very closely. The chemical space is quite large. It's a bit like, I try to represent this with the number that we have here. So it's something between the 10 to the power of 30 and 10 to the power of 60, which in the worst case is more particles than in the observable universe. So we need efficient computational approaches to maneuver that chemical space. And that said, you cannot use computational approaches to systematically explore that approach. You need some very good heuristics, some directions, some expertise to know where actually to look for this. And with this, I would like to switch to the actually core topic of this lecture, which is on the computers in drug discovery, chemoinformatics in particular, or chemoinformatics, because our field is very efficient, it's very small, so we haven't even agreed on a title yet. So some say it's chemoinformatics, some say it's chemoinformatics, but what unites chemoinformatics or chemoinformatics is that we're looking at chemical structures of molecules. We're trying to make the best use of that data. We connect it to data from other domains, like biology, to basically manage this data and to make the best use out of it. There are a few related disciplines, and sometimes there's a bit of a gray area in between. So we have bioinformatics, which is basically everything that is concerned mostly with the letters of the alphabet, or some subsets of these letters of the alphabet, and also protein structures. So it's a gene sequences, protein structures, and so on. Chemoinformatics is focusing on the chemical domain. Then we have molecular modeling, which is something that is basically mainly force field based. So it's basically we look into efficient representations of proteins and ligands and how they interact. And then there's computational chemistry, which basically tries to get down to the bottom of physicochemical properties by looking at quantum chemical approaches. So now I have been given 1 hour or so to give you an introduction to chemoinformatics, and this can feel to you like more like a roller coaster ride. I asked Copilot to visualize this for us. So what's going to happen is this, and you see the subtleties of hallucination. So Copilot gave me a beard, even though I didn't say so specifically. Chemoinformatics, yeah, there's another variant of writing it. And RD-Kit is apparently very important. And I would have guessed this should actually be a benzene ring, but never mind. So this is basically an impression of what's going to happen. So allow me to say, don't focus on the slides. The slides are mainly the background. Focus on what I'm trying to tell you a little bit of give you a bit of a sneak preview of many topics that will be presented and spoken about in the next couple of days. So let me show you a little bit what chemoinformatics can do. One of the very basic things chemoinformatics can do is help you with molecular structures. For example, understand subpatterns. So it's often challenging for people that are not medicinal chemists to talk with medicinal chemists sensibly, not to embarrass ourselves overly much if we are not familiar with certain terms. So there's a very nice tool from Matthias Rares' group who basically does pattern matching and basically gives you the terminology that allows you to communicate with medicinal chemists and actually work with them together. Because chemoinformatics has actually also a very important role in being a translator between different domains, right? So it's basically, if you can't speak the language of medicinal chemists, they will not follow your guidance. And this is one helpful tool. And actually, when you see the blue screen, the blue links, then this is a direct link to the website. So what else can we do with chemoinformatics? We can predict molecular properties because, as you may expect, many molecules have not been measured yet. And the problem with medicinal chemistry and drug discovery is that actually our compounds are fairly constrained in terms of where the sweet spot is in their physicochemical properties. So it's somewhere around here in terms of log P and molecular mass. So these are the two cardinal properties that decide mostly about the fate of a molecule, whether it can ever become a drug or not. And then there are many approaches out there. Nowadays, of course, very sophisticated artificial neural network-based approaches, but also very simple approaches that work quite well, like this one. It's like a phone book. It's like a lookup table, like an Excel sheet, basically, where you assign specific numbers, values, contributions to individual parts of a molecule. And then you sum them up, and you get a very nice estimate of the log P, which means the likelihood that you will find this molecule in a certain phase, like the water phase or a hydrophobic phase. So this gives you a very nice impression about the properties of these molecules. Now, what you can also do with chemoinformatics, of course, is you can assess the similarity of two compounds. And often, the Tanimoto coefficient is used as this metric. So this is just one of many different, but maybe the most widely adopted coefficient that is used to quantify the similarity of two compounds. And this here is a trivial example. It shows you basically how the two compounds, so this is acetylsalicylic acid or aspirin basically, and this is basically just a metabolite, so it's basically the acetylated form of acetylsalicylic acid. And here you see the similarity. So it's basically encoded as a bit string. This bit string is usually 1,000 or 2,000 bits, so it's usually use a compressed one. And then you can assess the similarity of two compounds with such a trivial approach. And nowadays, such approaches are still used. They are very effective. They are a little bit more sophisticated than what is shown here, but they are not of the world different. So once you have a possibility to calculate this Tanimoto coefficient, you can compare molecules very quickly. So for example, if we want to know if this molecule is in a database or whether similar molecules in a database, you can calculate the fingerprints and the Tanimoto similarity, and which gives you very quickly an overview of what are the most similar compounds and which ones are not. And actually, similarity is at the root of chemoinformatics. Chemoinformatics works, if it does work, mostly it works. If it works, then it works mostly because of the similarity principle, which tells us that compounds that are similar in structure, they will likely have similar properties. This is not always the case, as we will see, but often the case. So a lot of what I'm showing you today, so all these technologies on how to predict ADME properties, the QSAR modeling, toxicity, virtual screening, and so on, they all rest on this similarity principle to a large extent. Now, if you, as you imagine, a molecular space can be very diverse. I usually compare this, so basically the dimensions of the chemical space are extremely large. So basically when you get the new data set and you don't do some pre-processing or something and you want to digest what's in that data set, you may get lost or cry for help, right? And with chemoinformatics, help is on its way. Because it helps you, for example, to organize data, to cluster data. It's very nice shown in this example here. So basically, the slides have two properties today. The first one is to serve as a reference for later, so you can read the text later. But now, I will guide you visually through the concepts. So let's say we have these molecules. Each of these points are molecules. And then we want to know if there are groups of molecules, of similar molecules in there. The distances in between these molecules, these are exactly what we have seen before. This could be these Tanimoto distances or these Tanimoto similarities that are derived from molecular fingerprints. And we can now try to categorize these molecules and see if they cluster together. And the way this is done is iteratively just putting or connecting, linking the two nearest points. For example, A and B obviously are the two molecules that are most similar together. So they should be forming a cluster, right? And the next ones would be J and K, and then C and D. Okay, so we saw basically we just put always two groups of molecules together, which are the closest to each other. And now we come to a point where the distance between all of these pairs of dots or molecules is the smallest between two clusters that are already composed of more than one molecule. This is this one here, so B and D. So this means that these two subclusters are now connected. And this process is then iteratively going on. And we end up at some point with all molecules being in one cluster. So now, depending on the time you want to spend and the granularity in your analysis that you want to have when you look at these compounds, you can decide to use something like a threshold to show you exactly a clustering of compounds according to the threshold. So here we would have 1, 2, 3, 4, 5 clusters. If we use a lower threshold, basically if we allow less dissimilarity between compounds, then we use a smaller threshold. And the nice thing is, once you have calculated this, you can even do this at interactive speed, right? You can say like, this doesn't make chemically too much sense because I still see compounds that are different within one cluster, so I need to adjust my threshold. So this is actually very useful, and this is always the starting point when you work with larger sets of data. Another very nice point about chemoinformatics, and you will have a dedicated speaker on that, so Martin Sitcho will be talking about this, I saw in the program, is visualization. Chemical space visualization is extremely important to basically be able to translate difficult high-dimensional data into low-dimensional data. For example, this one on the right is something very trivial. It's a principal component analysis. But what it shows you is a reduction of a high-dimensional space into a low dimension one. So here we have only 2 dimensions, and what this graph shows you visually and very immediately, instantly, it shows you the relationship, the structural relationship between natural products, which are in green, and synthetic compounds, synthetically produced molecules. And it shows you at one instance that the natural product space is much larger than the synthetic molecule space. Okay? And that would be, of course, much more difficult to read from a table than it is from this visualization. So a lot of know-how and expertise goes actually in chemical space visualization. And with Alexander Warneck, who will be a speaker also at this conference, you have another expert on this domain in this workshop. Now, we have these billions and billions of compounds, and many, many more, realistically. We need to do something with them. What we want to do is we want to find the gems, right? So basically, we want to find new molecules that really have the capacity to ever become a drug. And the likelihood of one of these molecules in here being active is about 1 in a 10,000. So if you measure compounds in an assay, 1 in 10,000 molecules is active enough so that you can actually build on it, right? So it's basically, it's clearly active. It has a clear binding to the receptor. Realistically, you get many more hits because assays are often misled. For example, by assay interference, that's a big issue. But the genuine hits is about 1 in 1,000, 1 in 10,000. This depends very much on the target, on the druggability of the target. So what virtual screening aims to do is to basically reduce that haystack and give you a rank-ordered hit list of compounds among which there are nice ones. So it's basically like a hit list, right? In music, basically, you can do 2 approaches. Either you just pick a random music file and you won't like it, or you just look at the top charts and hopefully you like at least some of them. So this is basically an enrichment of interesting molecules that you would hope to get with virtual screening. And the nice thing about virtual screening is that most of these methods you can basically tune, right? You can exactly say what you need, translate this to the machine, to the model, and you get usually exactly what you need. So they are highly tunable and they are not expensive. So once these approaches are established, they can work very quick. So if you use, in particular, if you use pre-indexing methods, so methods that basically use an index, like you do Google search, they can give you results very, very, very fast. And one of these approaches that Thierry Langer will show you in full detail is the pharmacophore-based approaches. The pharmacophore-based approaches are one of many virtual screening approaches. But one thing that makes them really outstanding is the capacity to translate these models to medchem knowledge and to speak to medicinal chemists, because these models, the underlying concepts are very simple. So you can basically exploit this situation and speak to medicinal chemists that can then understand and follow your guidance. The reason is this: so most pharmacophore approaches, they use very few feature types. These are only hydrophobic areas or aromatic areas and hydrogen bond donor, acceptor, and so on. So very few features, 5, 6 different features. And this is exactly how medicinal chemists think. So if you go with this technology to the medicinal chemists, you will be speaking the same language. This means the translation into something that they do, like synthesis, getting a new compound based on your predictions. That's very simple. Another very powerful approach is this similarity-based approach. It's a shape-based approach. And what that means is basically, it basically suggests the following. If you have estrogen, which is the female sex hormone, so it's basically, if you're interested in finding a modulator with the same properties, you have 2 options, or at least 2 options. The first one is you do a trivial thing and you just look for any other steroid molecules, but the problem is that the low degree of innovation, right? So it's basically, what you would want to do is you want to make a jump, a leap, a hop. It is called a scaffold hop, right? So you want to come up with a molecule that looks entirely different. And the reason for that may be manifold. It's innovation, so it's patent protection. But it could also be that a compound that you need to jump on from has some toxicity issues or the pharmacokinetic issues that you might want to overcome. And this technology, pharmacophores too, but shape-based approaches in particular, they allow you to detect, to sense remote similarity. So the very specialty here is that you can find molecules to look similar in the 3D molecular shape space that are completely dissimilar if you would draw them on a sheet of paper. So you see, this is basically a famous example of two compounds. They look entirely dissimilar, and they have similar pharmacological properties, but the chemistry is totally different, at least from how we draw it. But the receptor feels the same shape, and this is exactly how shape-based screening works. So it uses these representations, and in this space, the molecular shape space, these two molecules are actually very similar. And that's a very nice thing. And depending, so basically in chemoinformatics, everything depends on the availability of data, right? So the more data you have, the merrier. So it always is a question of what kind of data do I have available? And depending on the answer to that question, you can employ different methods. And I saw you have very nice contributions in the workshop, in particular also to docking. When docking, basically the idea is how can I place a molecule in the binding site? This, of course, depends on whether such a molecule, such a structure of a target is known, or at least predictable, as you will see in the AlphaFold contribution and other contributions throughout this week. So it really depends on the data that is available. We can't do magic, but we can get close to it. We try to get close to it. So it all depends on the availability of data. And then, and Carl also pointed in this direction already quite nicely, the whole effort in drug discovery doesn't need to stop, or does not stop anymore at virtual screening. So nowadays, we do not stop at virtual screening, but we do the virtual screening, we expand it to target-based screening. So instead of saying, I want a compound to target this target, it's no, I want a compound that cures that disease. That involves at least one target, likely more targets, and it involves many biomacromolecules that I don't want to target. So instead of screening our library only against a target of interest, we would also screen it against all other proteins that could be of interest, or we want not to inhibit them, right? So nowadays, it would be computationally absolutely feasible to screen a library against a couple of thousands of proteins. It always depends on the granularity, on the available data, and so on. But this is the idea. So we do not stop at screening just on a single target. Beyond that, computational approaches can be used for many, many things. One example just here is metabolism prediction. I picked this example because we worked on it, and I picked it also because it works extremely well. It works extremely well because for the following reason. So this is basically a machine learning approach. And this is a machine learning approach. The aim of this machine learning approach is to pinpoint the atoms in a molecule where metabolism is going to happen. And the reason why this is so extremely valuable is because it tells medicinal chemists instantly where their compound may be too quickly degraded. And this is very important to know, because a medicinal chemist at all points in time needs to know whether the problem is overlapping with the pharmacophore. So think about, so the molecule has a certain size, and not all atoms in a molecule are relevant to the pharmacophore, so basically to the pharmacological activity. So basically, if this here is our pharmacophore, if this is the reason that our compound binds to the receptor, the medicinal chemist will always need to know whether this part is responsible for problems. Because if that part is responsible for problems, it will be very difficult to optimize this compound. Whereas if a other part that is maybe just for pharmacokinetics, for example, relevant, then there's a huge chance that we can actually fix this problem. So the medicinal chemists always want to know, what is the scale of the problem? You know, what is the scale of the challenge? Can I overcome it? And this is why these predictors are so important. And the reason why site of metabolism prediction, so basically pinpointing these sites, works so extremely well is because these are not necessary or primarily molecular properties. So in order to understand whether this is a site of metabolism or this one here, is we usually don't need to look at the whole molecule, but only at the proximate atom environment. And for that reason, the predictions are simpler because we basically look at only fragments, which are much more abundantly available in small data sets compared to other properties that are related to the whole molecule. And of course, there's lots of approaches out there for toxicity prediction. Toxicity prediction is one of the really the rising stars because in particular, environmental toxicity is something that people have not looked at until very recently. So if you want to have nice career perspectives, I would go to the interface, at the moment, I would go to the interface of toxicity prediction because this is really where everything points upwards. Carl will talk much more about structure prediction, of course, and there will be also further contributions. So this is now actually going a bit more into bioinformatics and structural bioinformatics and modeling. But nowadays, so this is just one slide to say nowadays, there's really lots of opportunities that allow you to predict the structure of target proteins. Now, the very important point here is, if you want to use these predicted structures for virtual screening, or as a measured structure, they need to be highly accurate, right? Because docking algorithms, they are very, very sensitive. They are also quite limited in their capacities, in their accuracy. So if you don't come up with an ideal model, the chances of success are fairly low. So you need to be aware of this, and you need to design your experiments accordingly. The reason for this is because proteins are flexible. In particular, so this is basically a cytochrome P450. It's simulated over just 100 nanoseconds, which is a very short time scale. It doesn't even show you any induced fit, so it doesn't even show any rearrangements that are caused by molecules that may enter the binding site. So here's the binding site, here's the catalytic ion here. So this molecule in particular, this is a nightmare to model. It's cytochrome P450 3A4. It's like an omnivore, so it basically eats whatever or chews up whatever you put there. There's even examples where this molecule increases the binding site by more than a factor of 2. It could even basically bind 2 drug molecules at the same time. So it's a bit of a nuts challenge to come up with compounds that don't bind there, because most of the drugs that are on the market do. Right? So it's very challenging. We need to always have this in the back of our head, that these molecules are not static as the X-ray structures might suggest to us if we just look at them not particularly carefully, but molecules are really flexible. And what's becoming increasingly more interesting, in particular also with generative models, and you will also have this part covered to some extent at the workshop very nicely, is the following thing. So far I was basically suggesting that we screen large data bases. That's not necessarily true anymore. So we keep doing that, of course, but another approach is to say, hey, I know a binding site, just generate me a molecule that fits this binding site. And thanks to the complementarity principle, you can do that. Because for molecule, in order to bind to this binding site, it needs to have compatible binding properties. This means if we have here carboxylic acid, we may want something hydrophilic, ideally a basic amine in this area. If we have here a hydrogen bond donor acceptor, then we will want a hydrogen bond donor here, and so on. And because most of these interactions have a direction and very specific geometric requirements, it allows us to build a molecule into the binding site. Right? So basically, if I only know the binding site, and if I know it, I can build a molecule into there. And this is actually one strength where machine learning is really pushing the boundaries in a way that we can evaluate these molecules not just in how they fit to the binding site, but whether the chemists will get gray hair if they try to synthesize it or not. So we can predict the synthesizability, which is a topic that I understand Alexander Warneck will present to you today. And it can also, at the same time, predict all the other properties. Are we still in the green with the admin properties and the tox properties? So the idea is basically to get all of these properties at interactive speed or at high speed predicted. So nowadays, it's no matter, I mean, no pharmaceutical company discusses whether they should use computational approaches or not. They are just a reality. They are embedded everywhere and throughout the drug discovery pipeline. In the early stages, to help us to really focus the experimental resources on the most promising avenues, and in later phases to really support making the best decisions about the very few compounds that get further. So computational approaches are everywhere nowadays, and it's no longer a trust issue. So it's basically that the most recent developments in the pharmaceutical industry are now that basically models are established for everything. So it's basically all the admin properties. You have these models, they are routinely updated every 1 or 2 weeks. So you have these models available. Now, until 2 or 3 years ago, the issue was still a trust issue. It's basically, it's nice, you have this model, tells me a number, but I don't really know if I can trust it. This is exactly the research that is going on at the moment. At the moment, methods are being established that allow us to estimate the uncertainty of prediction. So nowadays, a proper model should not just tell you yes or no, or good and bad. It should tell you how sure it is about this particular compound. Because it's all about the data that the model is trained on that defines whether a model will be, a prediction will be trustworthy for your particular compound of interest. Okay? Because these approaches do not extrapolate particularly well. So it's very important that you don't go away too much for the training space, or at least let the user know, be careful. Now we are going into a, in a domain, in a chemical space where I'm not particularly comfortable with. Chemists have no problem to work with something that is uncertain, but they need to at least know it. And this is exactly the most recent approaches or the most recent research effort that is ongoing in the industry at this time. So to work with molecules, so basically this was now basically a sneak preview where we will get to throughout the workshop progress of the workshop. But now, in the remaining time, I would like to tell you a little bit more of the foundations. It may not be so catchy, fancy, and super exciting, but it's the very foundations of that chemoinformatics or chemoinformatics builds on. And so basically, before we can do any of these fancy approaches, we can employ these fancy approaches, we need to actually know how to start or how we can use structures on a computer. So we need to first speak about molecular representations, because with a chemistry background, most of us think about caffeine in this representation. So it's a nice structure drawn on the computer or drawn on a sheet of paper. But the computer needs a bit more of a different representation to work with it. It needs to digest this structure as a number or as a series of numbers, as molecular representations. And what's is very much in the focus at the moment are the graph representations. With graph representations, where you present a molecule as nodes and edges that are connected, that represent the topology of the molecule, you can get very far. If you need some kind of information about the 3D molecular structure, for example, if you want to look at shape, 3D space properties, then you might want to choose force fields, like they are used in computer-aided molecular design very widely, which are very effective representations because it represents basically the molecule very efficiently as just spheres or even balls and springs in between. So it's very efficient. It's computationally and mathematically very efficient to describe molecules like this. And if you really need to go to the bottom of all the chemistry to understand materials, properties, reactivity, for example, then you may need to go to quantum chemistry, which at this moment, both of these areas in particular are benefiting greatly from machine learning, because machine learning can speed up things, in particular for the quantum chemical approaches, but also for the force field-based approaches. Not just speed, but also accuracy for the faster approaches. So molecular graphs. The big problem is, or the big challenge is, you need to get these molecules into a graph. And this is really a big challenge nowadays, because as you might know, all the literature, these nice PDFs that are very unuseful, if you look at it from a computational perspective, you need to get them into a representation, like a graph representation. And so a lot of research is now going into how to basically transform, or basically to detect the molecular structure, transform in something machine readable. And one of the free approaches out there is Decima from Chris Steinbeck's group. It's a really nice approach, where basically you upload a PDF and you get out the structures but interpreted. So in a machine readable format. So that's actually quite nice and maybe helpful. May come in handy at some point for you. So then how can we store that data? How can we basically represent molecules very simply? One very nice approach is SMICE notation. So the idea of basically formulating or representing a molecule in a single line. In a single line so that humans can still read it. And this follows a very few rules and basically tries to cover most of the chemistry that the molecule encodes. So it's basically follows very few rules. Basically the capital letters encode the elements, and oxygen is encoded as O, as you may have guessed, and C is a carbon. The single bonds are the most frequent ones, so they are not explicitly mentioned. So anywhere where there's no character is a single bond. The double bonds are encoded as an equivalent sign. And then you have parentheses that indicate branching. And with this, you have a very simple representation. You can go to the chemist, and then they can open something that is called like Excel for molecules, or something like this. Any program that can basically interpret such very simple representations, and suddenly the molecule is also in the screen. Because with chemists, you always talk with molecular structures. If you don't have molecular structure, you will be very quickly expelled like the water out of the binding pocket. And so SMICE, very nice. You can maybe try at home to come up with your own SMICE notation. So this is basically you just walk over the molecule, and then there's a ring opening, and these small letters, I did not explain them. So the small letters indicate that we are here in an aromatic system. And so complex molecules can be represented very nicely in SMICE. You can try to figure this out at home. Let me tell you just one thing as an outlook. There's one very nice descriptor, which is INCHI. And INCHI has a bit different angle. So whereas SMICE is something that you can still read, right? So if you have a little bit of experience, you can immediately say, this is, this here is obviously progesterone. So you might get there if you work a bit like that. But this one is then really getting a bit more tricky. So this is IUPAC's approach, and IUPAC has a different angle to it, because IUPAC is not working just with nice drug-like molecules. IUPAC wants a general representation of any kinds of molecules, also organometallic components and so on. So this one is a much more sophisticated representation. It's not necessarily human readable anymore. There are people that read that, but me not so much, not regularly. But the big advantage of the INCHI is that if you use the INCHI the right way, it can tell you immediately if two molecules are identical. And it can even be tau domain invariant. So it's basically, if you want to say, here is millions of compounds, make entirely sure that there is not a single duplicate in there, find all the duplicates. You know, some chemists write the tautomer like this, the others assign the other tautomer form. If you want to make sure that you would have all the duplicates identified, then INCHI is the way to go, because INCHI gives you a unique representations of molecules that can be tau domain invariant if you set it up correctly. So it's very interesting to know, right? So it's basically, whenever I have the question, oh, I'm working with lots of data, I want to know if there's any duplicate in there, realistically, you need to go via UPAC's INCHI. So this was 2D representations to basically try to cover the information that chemists draw in 2D space. But we can, of course, also look in the 3D space. In the 3D space, molecules can be very nicely represented with different molecular structures, molecular surfaces. They are explained here, so there's a different, it's like a bit of an individual dialect. It's basically how to basically translate what in reality is an electronic cloud into something that we can visualize as an electronic surface. So basically, depending on which of these dialects or representations you use, the molecules will look a bit different. So if you change software, like, if you're used to one editor and you look at the molecule and you look in a different editor, the molecule may look different to you, right? Because these representations are not always identical. So you need to be aware that depending on which viewer you use, the molecule may feel larger in the other viewer than it is in the other one. So there can be some differences here. And the nice thing is that with the 3D representations, you can also project properties onto the surface. For example, where are the hydrophobic areas in a molecule? And where is the molecule rather polar? Right? So this translates very nicely and gives you a nice overview of what the molecule properties are. What we now still need to know is how can we get some numbers out of that, right? So how basically can we represent molecules as numbers so that we can do some calculations? And these are the molecular descriptors. And until fairly recently, there was this book, like the Bible, which basically had represented all descriptors. So there are about 5,000 out there. A subset of them is useful. Another subset of them, maybe the research, maybe a reflection of the fact that we have many PhD students and they need to work on something. So there's a very high number of chemical descriptors out there. And even more so, you can generate many, many more with artificial neural networks. So artificial neural networks can produce learned molecular representations that basically you don't need to pick anything anymore. So until now, we often heard is the important thing is which descriptors are applicable to my problem. So I can pick the right ones to model my problem. Not so sure if this is still true, because to some extent, approaches are now smart enough to pick their own descriptors or even produce their own descriptors to answer a certain research question. But the traditional way of looking at descriptors is as a dimensionality. And I will just show you these two overview slides. They are a bit complex, but I'm trying to give you really the essence of it. So the essence is there are many descriptors, and they have a different information content. So there are very simple descriptors. They are called the 0D descriptors, and the 0-dimensional descriptors, they basically encode the presence and absence of certain atoms, for example. So it's something that you can simply count, ideally, or also for the molecular formula already. So basically, for example, how many oxygens do I have in my atom, in my molecule? How many carbon atoms do I have in my molecule? These are very simple descriptors. They are not very informative because there can be many molecules with 2 oxygens and this number of carbon atoms. The 1D is something that is already very interesting, in particular for example, for toxicity prediction. if I want to say this certain functional group, we usually don't like this in the toxicity context, or this is metabolically not stable, then you could encode this as a rule, as a substructure pattern, and this is basically what we understand as 1D properties. So these are somehow sequential information, but they don't use the full graph yet. This is the difference to the 2D descriptors. The 2D descriptors is basically the idea you generate one numerical representation, one scalar representation, or as a vector of the molecular graph. So you basically try to come up with one number per molecule. For example, is it lengthy or is it spherical? That would be a typical 2D descriptor. And the 3D descriptor is then something that is, again, very intuitive. It's just getting a number for the 3D structure, for example, for the volume. And we can then project also properties on this volume. And the 4D is another thing on top. It adds time, a time component. This means how does the 3D structure change over time? The next slide is trying to summarize exactly what I said here. And the following slides, I just added them, but I will not go through them in detail. Give examples of how these descriptors, what these descriptors look like. I want to emphasize on one thing. Now, naturally, if you think about it, the ligand and the protein, the approach is a three-dimensional process, right? So naturally, anything that is only 0D, 1D, or 2D descriptor, they miss a major thing because they miss the 3D component. Now, bringing in this 3D component can have value, but surprisingly, it's not always visible because the complexity increases so much that I may need to compromise on accuracy elsewhere. So basically, it's not automatically that the more you chip in data, the more you chip in complexity, the better the results. That's not the case, right? Cleverness is still the best approach. So you need to be smart about how to do that. Because when you work with 3D descriptors, you have the following problem. You have the problem that suddenly the ligand also is flexible because the ligand, of course, can adopt different conformations. What we see here, a molecule on the left is a molecule that you may be very familiar with. It's adenosine monophosphate, AMP. And this molecule has been observed to adopt very different shapes in different binding sites. So although these binding sites all bind the AMP, in different proteins, this molecule may adopt different conformations. And it is your challenge to basically represent these different conformations with conformations that are calculated because you don't know these for your compounds. Of course, you have not knowledge of the conformation of your compounds that you haven't even synthesized yet, right? So it's a challenge to reproduce it, to basically predict what the molecule will look like. And instead of working with just one representation, like one value, you're suddenly confronted with the need to represent this molecule in all different ways because you don't know which one of these conformations is relevant to your protein. I will show you this here in this example. So this is a molecule. If you want to encode this molecule, we can, for example, encode it as a very simple fingerprint and say, listen, I have a molecule of interest here. It has a hydrogen bond donor acceptor here. It has a positive charge group here and aromatic system here. But the problem being, this molecule is a bit flexible. So if you think about it, it can actually look like this. This means that these distances between these three properties, which I use to characterize my molecule, they change because now, in one of these conformations, this aromatic ring is up here. So all these things change. And so do my representations. So while here I say, yeah, I have this pattern. In the other conformer, I don't have this pattern, but I have a different one. So instead of working with just one description of my molecule, I need to look at very many variants. And you need force field for that. And the force fields are okay-ish to calculate, but they are not super fast. So you need to basically suddenly calculate with this formula here all the possible representations of your molecule. And the question is basically whether this really results in a benefit. Sometimes it does, sometimes not. And it's you as the expert to understand and know when it does and when not. So now we have a few minutes left, and I just wanted to show you where this travel is going. So now basically we finally in the position that we can translate molecules into numbers and work with them. And what we might want to do with those numbers is to predict properties. This is a very classical slide, so that slide could be from the 1960s or 1970s, although it would not be on a beamer then. But other than that, nothing much has changed. The idea is always, I have a target property. This can be anything. This can be toxicity, metabolism, target activity. And I want to have a model that basically predicts very well what I should have observed if I had run the experiment. Right? So this is basically what Qs are. Quantitative structure activity relationship or quantitative structure property relationship stands for. So the idea is basically to predict a certain biological or physical-chemical property. And here's a slide that just summarizes the many different properties that you could calculate. It's not a complete slide, but you see it goes all the way from physics, chemistry, down to really biology, toxicology, pharmacology. So exactly what we need. And the idea is always the same. If two molecules are similar in terms of structure, they will also behave different, same in these properties. So now the problem is, this is only half of the truth, because these linear relationships, as we saw it, just 2 slides before. So these linear relationship, this very simple linear relationship, these exist only for the naive. Because it only exists if I zoom in the chemical space really narrowly. Then I will see likely these linear relationships. But reality is different. Reality is often non-linear, at least on a larger scale. And this is a very famous example here that shows you that. So chemists have tried to optimize the compound on the left. And since it's a very small compound, they thought like, wow, let's grow it, right? Because this is what you do. You grow the molecules, you glue it somewhere to the surface. Chemists, trying to be very smart, and they are very smart, they added a phenyl moiety here. And look, wow, a huge increase of affinity. So now I need only 13 nanomolar concentration to reduce my enzyme, the reactivity, or my receptors activity by 50%. And so on. And so they tried also putting the phenyl moiety here. And I, ooh, my good, this also works. This is fantastic. So what do we do? Now, of course, we combine the two ideas, right? This is what everybody wants to do. And try to get even better. But the problem is here, suddenly, it's too big to fit, right? It doesn't fit the binding site anymore. This is something, a typical nonlinear problem. You cannot predict or solve this with a linear equation. So you need something that has a nonlinear function, because our molecules often show nonlinear relationships. And the way to do that, the simplest way, is to use a nearest neighbor approach. That's something very simple. So it's basically, if we are interested in predicting the green compound, right? So we want to know what is the property of the green compound. Then what we do is we look at the neighborhood, right? So what is the neighbor doing? It's like if you look at the test, you know, what's he writing, you should usually write the same, right? If you sit near to the next neighbor, to good neighbors. It depends on the neighborhood. But okay, so if I look at my 3 nearest neighbors, I would assume that this green sphere belongs to the red class. Do you agree? And if I would look at the 5 nearest neighbors, so I would also look at what the guys in front of me are writing. They're writing something different. So they agree with this person over here, or with this class here. So if I look at the 5 nearest neighbors, I may decide that this green sphere is actually belonging to the blue class. So here you see already 2 things. So first of all, it's complicated. And secondly, the answer depends on how I ask the question and how I parameterize my model. But this is a very simple approach, and often actually nearest neighbor is used still as a baseline to approach these problems. And so then we can use slowly more sophisticated models, although this one is not particularly sophisticated yet, but this is something that you may know from your childhood. So you may have, with your parents, walked out on a Saturday to go to the grass, to the meadow, and see some nice flowers, and then you happen to have this book which says, is the flower green or blue? Usually it's not blue because flowers are rarely blue, but you know, so you have this decision tree, and you might come up with the right name for your flower. And this is exactly what is done with decision trees as a model. So basically, you try to understand if a compound is a good compound or a bad compound. And then you can go through this decision tree. For example, is it too big? Is it very large? If it's very large, it's not a good compound. So one of these decision nodes could be, is it large? Is it hydrophobic? Yes or no? And if you then go through the tree, then you might come up with the right answer. And on the right, what you actually see is a real example of a toxicity predictor. So the tree can get as complex as you like. So for every flower, or for any flower on this meadow, you can have an answer. The problem is, if you make this specific tree for your meadow, it may only work on your meadow. If you then go on a trip to Thailand or anywhere else, this decision tree will not work, because by no means it will generalize. It will only work for your thing. And this is a general issue of machine learning. And one way to approach it is a very smart one. It's an old one already, but it's a very smart one. And it's called bootstrapping. So it's basically juggling with data. So it basically says one thing. Listen, I'm very limited in my chemical space, right? I know only very, very little about it. And I want to have the best possible exploitation of this data to use for machine learning. So in this way, what we can do is the following. We can select random subsets of the data. So rather than using one data and generate a tree, and that tree will be completely overfit to that data. So we say we use subsets, like usually 2/3 of the data, to generate individual trees. Think about this way. You have a company, and you want to hire medicinal chemists. If you hire these medicinal chemists all from one research group, they will have had the same teacher, the same books, and if you ask them a question, they may actually answer it correctly if they've learned it, or they will never answer it correctly, and none of them will answer it correctly because they have all had the same teacher, right? So what you want is you want to have one team member from this university, one from the other, and one from the other. And because it's virtual experts, you take 500 or 1,000. It doesn't cost anything to hire them. So you take 1,000 different experts, and the key point is all of them think slightly in a different way. They all talk chemistry, but they all have seen slightly different variants of the data. They have seen 2/3 randomly picked from that data. And they use also randomly picked properties of, in this case, molecules to make the predictions. And then what you have is 1,000 different experts that all tick a little bit different, and you ask them a question. And if you average what these 1,000 different experts say, then the prediction is much more likely to be correct. Do you believe that? So basically, if you have these classification problems or also the regression problems, with random forest you can get very far, maybe to 95% of what you can get with the most sophisticated neural networks. The only addition that a neural network has, it has even more applications. So it can do stuff that random forest cannot do. But as long as you basically just make predictions on individual molecules, random forest can get you really, really far for the reason that you have this very nice juggling of the data. And then, very last part, the artificial neural networks that everyone is talking about. So the idea of the artificial neural network is that they resemble the neurons and basically use inputs like descriptors or even generate their own representations and then give those basically these connections between the individual layers, the individual neurons. They get it some weight, and then they use what's very important. It's a nonlinear activation function to decide whether the signal should be forwarded or not. And then comes the final layer, which is the output layer, which then basically is the result of the prediction, which can be a yes or no, or basically a numerical value. So, and as long as you have 3 of these layers, it would be a very basic neural network. And as you get more of these hidden layers, the hidden layers are those that you don't necessarily have direct control on. As soon as you get more of these hidden layers, you're starting to talk about deep neural networks. As said, they have many interesting applications. For example, generative capacity. So it basically can really generate new molecules. This is where it's interesting, where it's particularly interesting. And yeah, and of course, this is basically the spearhead of research at this moment. Very last, let me just say one comment, is the modeling itself takes a lot of expertise, right? So it's basically, there's many steps that even you need to do until you can develop a model. So to be able to develop a model, you need to basically be the master of all the data. You need to understand the data. You must have clustered the data. You must have analyzed it, prepared the data. And only after having spent at least 50% of your time, of the total project already, you get to the point of model development. Then developing the model is like usually very fast, maybe 10 to 20% of the time. And then most of the concern is again related to does the model just try to treat me well, or is the model actually working in reality? And this is the final testing, or basically validation, evaluation. This is also where a lot of expertise goes in there. So exactly the point of the model development is rather the quick one, whereas the other ones, they take a lot of expertise. And nowadays, as these technical tools become more and more readily available, right? So basically anyone can do basic machine learning in very fast, very quickly, learn very quickly. So you see that the job market is changing now. The job market is no longer that you need only the machine learning expertise, because people have realized, hey, if you only do the machine learning, but you don't care about our chemistry, it's not going to go anywhere. So you see that now there's a shift back towards really chemistry expertise. So people really need to understand chemistry to really have a good prospect on the job market. So it's now the pendulum goes a little bit back into the chemistry domain, or quite clearly back in the chemistry domain, to bring really back together the computational approaches and the chemistry. And with this, I hope I was able to give you some of the key messages, some ideas of what you will learn much more about during this week. And I thank you very much for your attention. It's not the nearest neighbor.
Hello.
Hi.
So you said that the molecule can assume multiple conformations, right?
Yes.
So would that influence the clustering to calculate the distances between the molecules, or is it purely dependent on the chemical structures?
Very good question. This depends on what representation you use. Clustering is usually a fairly expensive approach, and usually you do it only in the two-dimensional space, not in the 3D space, because the additional information that you gain is not as much as you would expect. In particular, for example, you don't know the biactive conformation, right? You would only know it could adopt this bunch of conformations. So how do you compare them? You can do that, but the readout is surprisingly not much superior. Also for the reason because inherently in the 2D description, you have some 3D information. Think about an example. You have a molecule and you count just the number of bonds you have in between atoms. It will tell you somehow how far these atoms are away from each other, right? So you have actually some of this information in there. And to get beyond that is actually very unlikely that it really helps you to make a decision that otherwise you would have made in a wrong way.
Thank you. I would like to ask concerning interpretability. How does it matter in the machine learning you discussed? For example, in case of decision tree, you can always say that, for example, solubility is high because of some group being present.
Right.
In case of random forest, you are not, and for the neural networks, it's even worse. How does this matter?
Right, that's a very good point. So, so actually, I didn't talk about it because I ran out of time. But it's one of the key points is, of course, how can I learn anything from that? If like a fancy neural network or a random forest just tell me, you should choose this molecule over that one. I want to know why, right? Nowadays, there are approaches available that actually give you some insight, also in these very complex approaches. So speaking about machine learning being like just black boxes, I don't think this is any longer true. But what you do is you make a compromise, right? You say basically, I prioritize accuracy and applicability over the interpretability. The way I would answer that is the following. Think about, for example, toxicity, right? Toxicology is one of the most, I would say, inherently to the subject is one of the most conservative domains, right? Because you need to really be sure that you are not wrong with your prediction. That's why toxicologists really say, I want to understand what the model predicts. But even that domain has now realized the following thing. Actually, what I want to have is a correct prediction. And if on top of it, it can be interpreted, I'm even more happy. And maybe the right way to approach this is the following. To not say, I want the linear model because I want interpretability, or the other one. I would just say, nothing keeps you from doing a two-pronged approach, right? You can basically say, I take a weak predictor to have high interpretability, and for myself, for my in-house own use, I use a very sophisticated predictor that may not be interpretable, but it gives me the right answer, right? And then I need to still decide what to trust. And so I think the right approach to do is really like always fire from all, the guns you have, basically. You don't basically exclude one thing. Yeah. Thank you.
And we have a coffee break.
Okay, thank you, Carol. Thanks for having me. And as Carol said, I come from the Chemoinformatics and Nutrition Research Group from the URB. And I'm here now to sell you, free of charge, a couple of tools that we have developed in the lab, which are PDB-Cat and PDB-Cat, okay? So the first one, PDB-Cat, is a tool that is meant to classify and analyze structural data from the PDB. And it was developed by Ariadna, our PhD student. And as you can see, you have it on GitHub here. Actually, before the session, I posted on the Discord channel 2 notebooks that you can use to run these 2 software. So running them will take you a couple of minutes if you want to do it while listening. And I'll show you the notebooks afterwards. And in GitHub, we also have a notebook here that you can use, okay? So what does PDB-Cat do? As you know, the protein data bank is increasing its size exponentially. I also cheated here. I used Notebook LM, like I see everyone is doing. So with this exponential growth, we have more data. And well, Johannes said that before that this is great, right? The more data, the merrier, but it's also a handicap, right? Because we have more structures to process, more data to analyze. And this is where PDB-Cat comes in, right? So what does the program do? It processes the CIF files, MMCIF files from the PDB, which is the new format, and it classifies the structures depending on different things. The presence of ligands, okay? So it sorts the structures depending if they are in an apo or a hollow form. It also determines if the ligand, the biologically relevant ligand, is bound covalently or not to the structure. And it also identifies mutations if you provide an input FASTA files with your sequence of interest, right? So these may seem trivial tasks, but when you work on it, you see that not everything is as easy as it looks, right? Because, for example, for determining covalent and non-covalent interactions, it is actually a field in the CIF file that is not compulsory to fill, right? So there are covalent structures that they are not labeled as covalent, and this program solves that by calculating distances between ligand atoms and protein atoms and determining if the interaction is covalent or not by that distance. And in this way, what we found is that using a data set from the PDB bind, we identified 20% more, 20% more covalent interactions than the reported ones in the PDB bind, right? So this tool is also programmed to process data in parallel. So it speeds up the process, and you can specify how many CPUs you want to use. And in the publication, there is a particular example using the main protease of SARS-CoV-2. Okay. So this is a workflow that PDB-Cat uses to classify the entities in the MMC file. And so this will outline some features of the program. As you can see, it looks for entities in the CIF file, and it distinguishes if we have non-polymers, polymers, or branched entities. And in the case of non-polymers, there is a blacklist that the software uses to determine if the non-polymer entity that we're talking about or that we're looking at is a non-relevant ligand or is a relevant one. Okay, so the ligands that appear on that list are discarded, only keeping relevant small molecules. Okay, and basically for the polymer entity type, it distinguishes as well if we have long polypeptides or small ones. So based on a length threshold, we determine if that polypeptide can be a peptide ligand or is a protein. So it also identifies peptidic ligands as ligands. And it also classifies oligosaccharides, and it identifies glycosylations, which are covalent interactions that are labeled separately. Okay, so what we have is that from an initial set of files, the program creates two folders, one for non-covalent complexes and one for covalent complexes. So what I would like to do now is to show you the notebook itself so that you can run it yourself. Okay, so here it is. This is the example that I've posted in Discord, and it's a very simple example of classifying 10 main protease structures, right? So the notebook will first install the PDB-Cat, import required libraries, and then it will ask you to define some parameters. So here we define the folder name and this threshold for determining if a peptide is a ligand or not. So peptides with less than 10 residues here will be considered as ligands. And then if we want to classify the structures based on their sequence, we can also set this to true and provide a FASTA file of the structure that you want to check against. Okay. So then this here will upload your files. And this, so if you want to work with your files, you can put them in the CIF test directory yourself. And this is how the starting file structure will look like. So you will have this directory with the CIF files you provide, and this sequence that you can obtain from the PDB itself. And then the program does its magic. It will process these structures, look for the information that I told you about in the CIF file. And in the end, what you will get is an output folder with the classified structures. So the mutated structures, if you use the mutated filter, will classify on their own. And for the non-mutated ones, you will have subfolders for APO, and for the holo, you will have covalent and non-covalent, right? So when working with or starting a virtual screening experiment or campaign, the first thing that you will come across is how many structures do I have for that target? How many covalent? How many non-covalent? How many have mutations? And that program will quickly tell you the numbers when you provide the structure files. It also generates 2 data frames or CSV files in which you will have information for the targets and for the ligands. So here is the information for the target files. You have 1 line for each target and some information. And something that's interesting, in the ligand table, you have 1 line per ligand, and in one of the columns, it will annotate if it's a covalent ligand, if not, it binds covalently or not, and the covalent bond with the actors of that covalent bond and the distance that the program has identified. And finally, this notebook will download the results for you. If you don't run it in Colab, you don't need it. So that's it for this tool. And now what I would like to present you is the other tool, which is PDB-Cat. And this tool is developed by Said. He's soon to be bachelor degree, which has worked in our lab and is here today presenting a poster about that. So you can go ask him for details. And this library, what it does is analyze interaction files, right? So you can use programs such as Arpeggio or IChem to obtain the interactions in a given PDB file. And this is a mess. It's just this. It's a mess of JSON files, endless JSON files that have all the data, but it's not human readable, right? So what this program does is collect all these JSON files, all this input, and it analyzes, incorporating also bioactivity data, okay? And it also provides functions to filter and sort the data according to your liking. And finally, it can generate also plots for you, which are interesting, well, for publications, but also to understand the data, right? So what you end up getting is that you have a lot of files of raw data, and it creates a matrix that incorporates the essential information there, which you can use to produce the plots. So this is how the JSON files look like. And yeah, hard to read. And finally, through that matrix of important interactions, you can use the plots that the library provides, or you can use the matrix to generate the plots yourself. So I will show you the notebook now. And again, it installs the library. And here I download the data. And this is how the input directory looks like. So you will have the JSON files that you have obtained with Arpeggio, for instance. You can also provide a file with which ligand belongs to which subpocket, so that you can use it to color your plots. And this is the activity data, so the activity that each ligand has in each structure. So the program will first process those JSON files and generate you this matrix, okay, in which you have the essential information for each structure. You have each structure in one column, and here is the type of interaction and the atoms that take part in the interaction. Okay, and this is what the program finally uses to generate the plots. So after running this code, which is long but not that slow, it's pretty fast, you can generate these plots. For instance, here we have the number of intermolecular interactions with each residue, and as you can see, you can color the different residues according to the given subsite, as I showed you before with the subpockets file. You can also see in these plots which type of interactions are more frequent for each residue. And the cool thing is that this is all automated, so you could take a target, run it here, and have a quick exploration of what's happening. And one interesting feature is these heat maps, in which you have the different type of interactions on the left and the residues on the bottom. And you can plot these heat maps with the property that you want. So here, for instance, we're looking at the mean activity, because we linked activity to the program. We look at the mean activity that the ligands who have a certain interaction with a certain residue have, right? But we can also plot the counts. We can see how many structures have that interaction, okay? With percentage, etc. So what we have is that for a quick exploration, it gives us a lot of information. For instance, if we compare the counts of interactions with the activity or the average activity that we get for each interaction, so that we can see that some structures occur, or some interactions occur in a lot of structures, and therefore we can consider them basal for activity. But that does not mean that we will get increased activity for making them, right? And we can identify interactions that do not occur as much in the PDB structures that we have, but for those complexes, we have high activity of the ligand. And therefore we can, for example, this is an example run on MPro, and we can identify, for example, this hydrogen bonding interaction, which occurs in most of the complexes, so it's required and basal for activity, but does not mean that the compound will be active. Or we can identify interactions that occur in a fewer number of complexes, but are interesting because the ligands are more active, right? And particular subpockets, etc. So that's all. Thank you for your attention. And I invite you to try these tools yourself, because they are really easy to use and almost with a click, and I hope they are useful. Thank you.
So very nice and useful tools, I would guess. And my question goes to this calculating distances with the covalent inhibitors. So how, what do you use under the hood for the distances, and how fast is this, like in terms of number of complexes, CPU, parallelization, GPU, etc.? Thanks.
Yeah, thanks for the question. Maybe Ariadna is better suited to answer this, but the quick answer is that yes, it's really quick, and but I can look it up and answer you. Actually, with this tool, you can process the PDB in a matter of hours. So.
Hello, I have a question. I just have a curiosity on the workflow on how the entries were classified in the PDB classifier. So the PDB-Cat, on the this one. So in the why you only consider only polypeptides and proteins? Aren't nucleic acid interesting?
Yeah, so I didn't go into it, but this would be here, right? So the polymers that are not polypeptides, they are not classified as ligands.
Okay, so I'm just saying, you know, RNA and small molecule bindings to RNA, it's not possible with this.
No, that's true. Yeah, it wouldn't classify them in another folder.
Okay.
But if that's something that's interesting, of course it can be implemented.
So basically, if I run, I just have to go to the other folder and then inspect the classification myself.
Yeah,
okay.
that might be possible. Yeah.
Okay, so I would have maybe last question. If I assume, well, so you need the PDBs to find first, or you need a sequence, but you assume this for the, let's say, families which have a lot of frequencies in PDBs, but you have, if you consider the fact that you have in history the PDBs done on mice proteins or bacterial proteins, and my sequence homology is like 80%.
Mm-hmm.
If I would put the sequence and you would put the cutoff for the identity, does it do it? I mean, do I have to find it myself, the PDBs, or the PDB-Cat does it?
It does. So the way that works, you can also provide different sequences, and anything that is the exact same sequence will be classified as non-mutated, and the rest will be classified as mutated. But in the output file, you can see if you specify this mutation or several sequences, it also calculates, right, how many mutations you have. I can increase that for you. So, for instance, this structure contains 12 mutations respect to the sequence that you passed, and it passes you it gives you the mutations, and it also calculates the identity percentage here.
Mm-hmm.
So you can sort of get that from the output. So
this is when you submit the PDBs.
Excuse me?
This is when you submit PDBs, but this is if you would submit just one sequence, can you submit more sequences, or is it because the question is if you would submit one sequence, I put cutoff, or if I submit one PDB and I would give a cutoff in RMSD, I would give him 0.6 angstrom in RMSD and find me all the PDBs you can find.
Yeah, so this is you do not submit just a sequence and then it gets the PDBs. It's not the way it works. So you have to submit both, right? So you submit the structures,
yes,
and then you submit the sequence of your interest.
Okay.
So yes, you can just input the structures that that belong to the sequences you're interested with, and that's it. Thanks.
Okay, I think you actually can go.
All right. Well, thanks so much, everybody, for giving me the chance to speak here at 980D. I'm sorry I can't be there in person, but I'm really honored to be able to contribute to this. The subject of my talk, I was asked to speak on Rowan, which is the company that I co-founded, and sort of this paradigm of easy online access to molecular modeling tools. So what I'm going to do today is just sort of talk a bit about Rowan and sort of the paradigm we're trying to push forward, and then spend most of the time just going through the various tools that we've developed or host through Rowan, and then some of the science behind that, with an emphasis on how it can be useful in drug discovery. And then tomorrow, my colleague Jonathan will give a tutorial on some of how to actually use Rowan for practical matters, like a hands-on demo. So just briefly, can everyone hear me okay? All right, I'll take the silence as success. Just briefly, by way of background, I originally studied at MIT, where I did experimental. What's up?
You can continue.
Oh, okay. Sorry, I thought someone was trying to say something. Forgive me. So my background is in experimental chemistry, actually. So organic synthesis, medicinal chemistry. I did antibiotic drug discovery research back in the day. I then did a PhD in organic chemistry where I was about half and half experimental and computational, focusing on organocatalysis and sort of other related matters, high throughput screening, computational modeling. And then since then, I've been working at Rowan for about the past 3 years, where we're trying to build sort of modern scientific tools that are more accessible, I'd say, to the general scientific populace. And so sort of to get into what we mean by that sort of more concretely, we can think about, sort of at Rowan, we think about computational chemistry as having maybe 3 eras. So the field is not that old. Organic chemistry, obviously, is a very old field, centuries, if not older. But computational chemistry has only really been around in a modern sense since the 1960s or 1970s, since the advent of sort of mainstream computing. And back in the day, if we think about the early days of programs like Amber or Gaussian, this was done, in sort of mainframe style computing, where you would go, there'd be a centralized computer for the university, maybe, one computer for a department or for an entire school. And then you would timeshare, you'd use time, you would input commands there, and only a very, very few people had the expertise needed to do this, right? So it's very, very expensive. Very few people could actually participate in the work of computational chemistry. And these early studies they're beautiful to read, but it's not diffusing out into the world, right? It's not having the full impact it can have. People in everyday drug discovery programs, whether academic or industrial, are not able to use these techniques at all. I'd say sort of the current era we're in is what we think of as the desktop plus high performance computing era, where there's much less expertise needed, right? So now we have graphical user interfaces. Many more people are able to do computations. The total cost is much lower. So you need a HPC cluster, but you don't need a dedicated the computer for the university, because computers have gotten so much more powerful. But sort of at Rowan, what we think is looking at other areas of software, what we want to push is sort of this idea that we can do this all through the web, that we can do this through the web. We can make it just like Gmail, Google Docs, Uber, Airbnb, sort of all of these other applications that are just everywhere in our lives. For scientists, we think that computation should be right next to you, sort of in that process. And our goal is that this can both bring the cost down of computation, so we can make it more cost effective and like just cheaper for everybody to do more computations to get more scientific insights, and that this can mean that more people can do it. And so, obviously we're not there yet, but the dream is that, one day there won't be, chemists who don't do at least some computations, just like everyone who, writes a book now does so in Microsoft Word. You're not a computational author. You're just a regular author who uses computation to help you, who uses software. That's sort of our hope, is that, in the future, even, the best medicinal chemists will still be using a little computation to help just supplement their insights, and get more, do the things that are hard for humans to naturally do, like reason in 3D, predict binding poses, and all of this stuff. And I think this has been a dream of the field for a long time. So this is, if we think back to 2007, you can see the distinguished drug discovery veteran John Van Dree writes in a perspective that like, maybe in the future, we'll take computer-assisted drug discovery tools and put them onto the desktops of medicinal chemists. That was a dream. And then he goes on to write, I'll just read directly. Too many modelers are still in the business of being docking slaves for their experimental collaborators. The experimentalist asks the modeler, please dock my new idea for a molecule, and waits for the result to see if it confirms their design. And so we see that there's this sort of division of labor that just doesn't make sense, that ideally the person with the idea will be the person steering the computation. And so that's what we're trying to do with sort of these easy access to modeling tools, that we'd like anyone who understands chemistry should be able to use software to do that. People doing drug discovery need to be experts in drug discovery, but they maybe don't need to be experts in programming as well. We think that, as we break these barriers down, it will be good. It will promote sort of understanding between computational and experimental scientists. And frankly, we think that putting this all on the web will be quicker to update, cheaper to operate and maintain, and just simpler for the end scientist. It's sort of how other areas of software have gone, and we think that's where this field is going as well. And that's just a brief sort of like vision statement for this idea of easy access to online modeling tools. Just a couple facts about Rowan just before we get into the meat of the presentation. We're a startup founded by me and two other people in 2023. We're based out of Massachusetts. We work across a variety of scientific fields. So I'll be focusing today on small molecule drug discovery, which is where most of our work is. But we also do some work in enzyme engineering, materials science, homogeneous catalysis. We have almost, over 8,000 users, scientists who trust Rowan to power their work and cite us in lots of publications, running millions of calculations. And yeah, we're just getting started. So we're excited to be able to share a little bit about this with you today. Okay, so what do we actually do? What are the tools? What can you do through Rowan, and how can this maybe be of interest to people who are thinking about drug discovery and accelerating that with computation? One of our sort of fundamental paradigms at Rowan is that everything should be doable both through a graphical interface and through code. That when we ask scientists to choose between these two paradigms, it's usually the wrong choice. And that's because on the one hand, when we only do things in code, when we only see data in tables or CSVs or dumps, we don't get the insight that we need as scientists. So there is a sort of there's a unique sense you get from actually examining a molecular structure, of looking at a protein or a ligand or a conformation or a pose. There's something there that to date, we think that we can't capture through text, that you can run all your automated checks there, but when you actually look at your data, you learn something special. On the flip side, one of the advantages of doing computation is that you can do a lot of computations. And so when we do everything only through graphical interface, it quickly becomes tedious. So sort of the dream is that we can interoperate between the two paradigms so that we can say, I'll run the calculations, I'll run one to make sure it works, then I'll run 100,000 of them. And if any data points seem bad, I'll go back and look at them again visually to sort of check and understand what's going wrong. And so through Rowan, we have essentially two ways to run every calculation. We have the way to do it through the web by clicking without writing any code. And then we have a way to put it all into Python so that you can put it in an active learning loop or run it 100,000 times or any of this stuff. But the science is exactly the same, and all the results are saved to the same place. The way that we generally think about how to approach this problem is as building out a toolkit for molecular design. So this is just a piece of classical art, or this is Christ in the House of His Parents by the British painter John Everett Millais from 1850. And I just chose this because it's a carpentry shop, and you can see there's lots of tools. So there's a saw, there's this ladder, there's lots of little awls, or I don't exactly know what all those tools are back there. But the point is that for every occasion there's a proper tool, and that there's not one task in drug discovery which can solve every problem. That you sort of want to have a nice like a bucket of things that you can pull out based on where you are in the program, the nature of the target, the nature of the problems you're encountering in the lab, and so on and so forth. And so, building the most useful software looks like having a lot of tools, not just one really, really good one. If all you have is a really, really sharp saw, you could do a lot, but you can't build a door or a table. And so this is sort of just a visual overview of like the tools that I'll talk about today, and sort of where they fit in the preclinical drug discovery landscape. This is broad, and I'm sure people here can critique exact positioning of these or say that this is incomplete. This is meant to just give an overall idea. So early on, we often have sort of like these hit discovery or high throughput virtual screening. Then you take your hits and you sort of expand around them. You do some hit to lead and then maybe some lead optimization. And the nature of the problems as you go from looking at many sort of weak binders to a few, hopefully strong binders that you need to think very carefully about what their in vivo properties are going to be. The nature of the questions you ask tends to change. And so we have sort of different tools that we recommend to our users as we do that. So sort of the first, I'll divide this up into 3 categories of tool just for conceptual clarity. And the first bucket of tools are what I think of as just structure-based drug design. This is sort of what people think of when they think about, using computers to help discover drugs. So one of the first thing that we do, sort of when we think about top of the funnel, this is where we consider what we call batch docking. And this is docking not where you have a specific binder and you're really trying to understand the pose and like design these interactions. This is docking where you say, I have a big list of compounds. I have a target with a pocket that I know. I'm just going to throw a bunch of compounds against it and see what sticks. And so we've built out a high throughput pipeline at Rowan. We've tried to do a good amount of hardware optimization and all this to get this as fast as possible, just for the task of saying, let's dock this all as quickly as possible. We're not going to run a ton of analytics on these as we go, just to keep it fast. We can always go back through and redock them later if we need to. This is just about enrichment. Can I pull hits out of a library? And so what we've done is, you can see this is sort of what the interface looks like. We have a list of molecules we've docked. We can see the target in another place, and then we just have a distribution of docking scores. We say the lower is better, and so what we hope is that the compounds down here with the lowest docking scores will be, at least enriched in true binders. They won't all be binders, right? Docking lies to you a lot, but there will be, meaningful enrichment here of true binders. And when we actually check this scientifically, we're not the first people to do this. Many, many other scientists have benchmarked these sort of high throughput docking workflows. Folks like Brian Schoikid at UCSF and many others, they've done fantastic scientific work here that we're just following. We see that this can actually work. And so if we take one of these, this is data as the MT1 target benchmark from the DUDE-Z set, which I forgot to put the reference here, but I can send it to anyone who wants, or I can easily find it. When we sort of run this benchmark, we can see, we're able to get meaningful enrichment here. So ideally, here we're seeing that the true positive rate is higher than the false positive rate. If we look at just the bottom, the highest scoring 0.5%, we see about a 14-fold enrichment of hits relative to a random sampling. So it's, it works. It's not amazing, but this is about as well as docking works for these targets. And what's exciting for us is that with sort of the hardware optimizations we've done and with the careful sort of like use of multiple processors and load balancing, we're able to get this down to about 1 second per compound, a little bit more, using AutoDock Vina. And we have other programs like QVina that we support that can go a little bit faster, that shave a little bit on accuracy off here. So it's it's something that you should benchmark for your system. But this can easily get you to tens of thousands of compounds to millions of compounds if you run it on multiple machines. If you do want to do billion scale docking, we don't currently support that in Rowan, but the best way to do that is to build an active learning loop atop something like this. So dock, 100,000 compounds, build an ML model, and then iteratively run more rounds of docking to improve that. So if we now imagine we sort of have a hit here and we want to zoom in, Rowan does support this sort of like classic, glide style docking workflows where you visualize the pocket and the ligands. You here have overlaid a bunch of poses from redocking this PDB to GQG. And you can see that there's a lot of docking scores by conformers. And we've also added these other analytics here. So the user can control this, of course, but we enable you to run both ligand strain calculations, so identifying how strained the pose is in kcal per mole, and also these pose busters calculations here, which essentially just say, is this pose physical or is it garbage? The strain idea, I think, is quite a fun one. So these authors, Matthew Smith, and Shuo Gu and coworkers, basically found that you can sort docking poses and you can get a pretty good filter for if they're physical or not by just looking at what the strain of the predicted bound conformer is. And if you say this is a 10 or 12 kcal per mole distortion from the ground state, it's probably not going to be the way the molecule binds in real life. Just in practice, you can imagine a scenario where that's technically true if you have very, very strong, salt bridges or something. But in practice, it seems to be a quite good filter just to use a strain calculation and see if this pose physical or not. And so what we've sort of done in Rowan is we barcode it here. So we say below 4 kcals is fine. That's sort of what you typically see. 4 to 8 kcals, yellow light, this maybe you should look at this pose carefully to see if it's real. And then above 8 kcals, we typically say, we're not predicting that this pose is physical. There may be some flaw in how we're doing the calculation, or the structural model may not be perfect. So, we can't say for sure, but we would advise you probably to treat this pose with a lot of caution. And this is just sort of another check on the, I'd say, finicky nature of docking workflows that you may have experienced before if you've done a lot of computational drug discovery. We also do blind docking. So if you don't know what the pocket is, you can dock sort of against the whole protein. Here we just rediscover the same known pocket, but in some proteins there are meaningfully multiple pockets where something could bind, and so you can actually discover potential pocket sites there. And we'll be adding sort of a specialized pocket detection workflow to Rowan sometime in the next few months. Here we use QVNW, which is a highly optimized sort of blind docking code. So this runs at about the same speed as regular docking. And then, of course, we run co-folding. And I believe there's a lot of other people talking about protein folding at 980D, so I don't want to go too much into how this works here. But just to say that this is very much becoming, I'd say, a complement to docking in how these things are often done, sort of in small molecule drug discovery. So co-folding gives you different things. You can account for changes in the protein much more easily than in rigid docking. It's also somewhat slower. You get, if you run bolts 2, you can get these affinity probabilities and IPTM scores, which are different than docking scores. Sometimes seem to be better, sometimes seem to be worse. But it's a useful thing in the toolkit. And I think how well it exactly works depends a lot on the series. The thing that I just want to underscore with both of these before we move on is just to say, validating all of this is really, really important. And when you go talk to people in cutting edge biotech or pharma companies, this is the thing they emphasize too, I've found. It's just that all of these methods can have issues and can lie to you. And so the way to do careful drug discovery is to try multiple approaches. I say check the obvious things. So check that the stereochemistry is actually right from co-folding. We've added automatic checks for that in Rowan because it doesn't always get preserved. And so you might dock the L enantiomer or you might run co-folding thinking you're co-folding the L enantiomer, and bolts 2 will give you back the D enantiomer, which is frustrating. So, check these things, check the strain, check the confidence, use pose busters, and then do sort of an orthogonal check. So either MD if you're in silico, or if you could get an experimental crystal structure, obviously that's even better. And we have added support for protein ligand MD straight through Rowan, so you can just hit resubmit on any pose. It'll run MD trajectories, and you can look at a lot of things here. So you can download the PDB and do whatever analysis you want. You can look at, various residue contacts and all this stuff. But typically the first thing we look for is is the pose stable? Because you can see here in 4 replicates, the RMSD of the ligand to sort of the protein structure here is shown to be very low. So, whether the pose is exactly what you'll see experimental or not, it's not immediately exploding and floating off into space. And that's, I think, a useful physical check that is very complementary to tools like protein ligand co-folding models. That sort of concludes my structure-based drug discovery thing. I showed free energy perturbation on a previous slide, but we'll get to that at the end because that's something new that's coming to Rowan. And I want to focus now on sort of what I think of as classic simulation tasks. And these are maybe a little bit less flashy than structure-based drug discovery. So it's immediately obvious when I show you something like this. I think what the relevance to a drug discovery campaign is. You're literally seeing a target and a ligand bound to each other. It's not hard to imagine oh, this must be really useful. I think when we talk about sort of these more classic simulation techniques like conformer searching, it's not always quite as directly obvious how this helps people make new medicines. But I think the point to underscore is that this is often in a project where you start really asking tough questions like, what is going on here? You know, maybe you're stuck. Maybe you're trying to find a way around just an activity ceiling that you can't. And this is where, in our experience, people start to really, really want to understand what their compounds are actually doing and how they're behaving. And so this is where we turn to what I'd call sort of higher fidelity simulation techniques like quantum chemistry. I think one of the things that we get a lot of use for in Rowan is just running conformational searches. So we support users running conformational searches through a variety of methods for generating conformers. So we, through CREST style like iterative metadynamics, through just RDKit like torsional distance geometries, through Monte Carlo simulations, through diffusion models. So you can generate conformers a bunch of ways. Then you can screen them all with a bunch of levels of theory and deduplicate them. And what you get out is essentially just a conformational map of how a given molecule behaves. So this is dasatinib, and you can see here we've overlaid the lowest 10 conformers. You can see the predicted Boltzmann weight of each conformer. And then you can see sort of the properties it has. So the solvent accessible surface area, the polar solvent accessible surface area, and the radius of gyration, and sort of obviously the energy difference in kcals per mole. And then what you can do, so this is done using one method, so you can add a methyl group and see how the conformational landscape changes. You can run it in chloroform instead of in water and see how the conformational landscape changes, and sort of build up your understanding for how this molecule will behave in different environments. One of the things that we recently added, which I'm pretty proud of, is the ability to see quickly sort of how the conformational ensemble's energy varies as a function of different internal coordinates. So if you want to understand cis versus trans amides, which is a pretty common preference, you might actually just highlight these atoms in the viewer and say, show me all the conformers and show the energy on the y-axis and then this specific dihedral angle on the x-axis. And what you can see is that at least in this search, the cis amide is predicted to be favored, which is a little bit of a surprising conclusion. And so we might want to look and say, what did we start with? Are we running a thorough enough search? Is it really true that there's only 5 trans amide conformers here, or am I biased? Or am I missing some part of the landscape in some way? And here I ran this quite quickly, so I think in this case that's almost certainly the answer. And then finally, you can always go and sort of look at the ensemble average properties here, like the average polar solvent accessible surface area. And that's also nice sort of as a single metric to compare. If you're doing, linkerology and you're trying to understand how different substitutions might impact, the behavior or solubility or permeability of a molecule, it's nice to be able to get a single value out so that you can compare along a series. One of the first things we did at Rowan actually years ago was working on pKa prediction. And it's been something that we've spent a good amount of time thinking about through the years. I show this slide because I think the difference between different ways to predict pKa it's very important, and I think remains a little bit confusing for people. And so this is the clearest way I can think of to explain it. If we sort of take as our reference neutral microstate, this gray ball here, considering that each ball is a state, and we'll say the blue states are, anionic, the red states are cationic, and then the gray states are neutral. We can predict either in the microscopic or the macroscopic paradigm. And the microscopic paradigm essentially just looks at single permutations. So if I protonate a single position or deprotonate a single position, what will the pKa for that change be? The macroscopic pKa sort of looks at the overall charge transitions. Like if I'm running a titration on an ensemble of molecules, what will happen? And I think both are very valuable. So one is not better than the other. This one is localized, so it tells us about individual functional groups. Microscopic tells us about localized changes, and then macroscopic tells us sort of how the ensemble of states will behave. And one of the things that we've worked really hard on is making these pretty easy to go back and forth between. And I like using this example of piperazine just because it really showcases the differences quite nicely. And so if I look at piperazine here, and I ask what the pKa of these two amines will be, I get back from my microscopic predictor that they're both going to be about 9. So this is 8.99, this is 9.09. These are virtually the same. This just shows you the numerical noise in the calculation. And then I get this little warning here, which says essentially, hey, these are like almost degenerate. I think maybe you need macroscopic pKa here. And then indeed, if we go over to the macroscopic pKa view, we see that the predicted overall charge transitions actually match experimental much better, because we have, essentially, we see that we should get one protonation event at around 5.6 pKa units, and then the other one at about 9.5 pKa units. And so this actually, if what we're trying to mimic is an experimental titration measurement, this actually shows us what we'll get much better, because we'll get, if we're at neutral pH, one will be protonated and the other won't, obviously because there's an inductive effect between the two. And so I think for most drug discovery applications, this is probably the first place to start, is with a macroscopic pKa prediction. But it's worth remembering that, this now actually does represent sort of how hard it is to protonate each one. And so that can be useful as well. I think they complement each other. Macroscopic pKa predictions obviously blow up in complexity very, very quickly, sort of as a combination of the number of individual proton transitions. And so we've done a lot of work, and this is a machine learning method where we essentially learn a free energy function over each microstate. And we try to prune the microstate ensemble so that this doesn't take more than a minute or two, even for very large molecules with a lot of states. We have a whole, it's documented in this paper, but we have a whole beam search algorithm that we use. But what's cool about this, now that once we have the pKa ensemble, we can do a lot of things like sort of predict pH-dependent solubility. And so we can show that, for a molecule like this, the water solubility at basic pH is predicted to be very low. But for instance, in a stomach environment, this low pH, this is predicted to be very soluble as well. Moving on to sort of like just the bread and butter simulation stuff, you can do quantum chemistry of many sorts through Rowan. So you can, plot orbitals, you can plot electron density and potential isosurfaces, like you might see in a textbook. You can do reactivity modeling. So this is a, benzyl bromide plus cyanide SN2 reaction that we're viewing with a scan. And we've recently added support for, double-ended transition state search methods like string methods, so this is a freezing string calculation where we're looking at this bioorthogonal reaction between a strained cyclooctine and then this methyl azide. And so this is all documented, I think, pretty well, and we're quite proud of this sort of quantum chemistry area of our platform. One of the things that we've worked really, really hard to do is make it so that many, many different levels of theory and sort of different paradigms for chemical simulation are accessible through the same interface. And so if you want to run density functional theory, you want to compare it to maybe Stefan Grimme's newer semi-empirical methods, and then you want to compare that to a neural network potential. This is all possible to do through Rowan, essentially, just by changing a toggle or by choosing a different method in a select box. And I think people don't maybe realize, who haven't done a lot of quantum simulation, how different these things are. So this was a benchmarking study we did where we compared a lot of different methods of theory on evaluating strained conformer energy. And you can see that, your choice of method determines almost 30 kcals per mole of accuracy and almost, I think 13 orders of magnitude in time. So calculations can take, milliseconds, or they can take weeks. And these are sort of things that historically require very, very different software infrastructure and software packages. And so we've worked really hard to make it as easy as possible for the end user to choose the level of theory that makes sense for their task, without having to learn a new package every time they want to switch from, GFN2 XTB to AIMNet2 to R2Scan 3C, that they can do it all just with the click of a button. I want to zoom in just for a couple of slides on the neural network potentials, because this is one of the fastest moving areas of computational chemistry right now, and I think something that people should know a little bit about. It's actually pretty old. So Michel Parnella published the first high-dimensional neural network potential back in 2006. And the idea here is you essentially just train a neural network to learn the potential energy surface. So directly mapping atomic coordinates to energy values, essentially without all the machinery of quantum chemistry or a force field. The neural network just does it all internally. And this seems like it shouldn't work, but it turns out it actually, in the era of big data and lots of GPUs, it actually works frighteningly well. Sorry, this is just saying the thing that I said in words, again, with diagrams. So with a neural network potential, we're essentially just removing all of the wave function machinery of quantum mechanics and just replacing it with a single pre-trained potential. We didn't believe this at first, so we read the papers and we started trying to do our own benchmarking and applied studies. And so we've published, I'd say, a good amount of work in this area over the past 18 months. So PK prediction, benchmarking on conformers. We released our own potential, benchmarking on BDEs and redox potentials. And I think right now we try to keep eyes pretty closely on the field. And I think if you have sort of a routine drug-like molecule, your first choice for a simulation task should be a neural network potential, I think. And one of the areas that has just recently become possible with this, I'd say in the past year, is predicting bond dissociation energies. So we put together a database of experimental BDEs. We showed that essentially NNPs could reproduce high-level quantum chemistry almost perfectly, with the exception of acetone, which is actually a failure in the DFT. And that we do this, we get many orders of magnitude speed up. So this is accuracy here. This is time. So where you want to be is at the origin. And what we can see is that the green dots here, these neural network potentials, essentially have the same accuracy as density functional theory. They don't have the same problems as semi-empirical methods, at least historically have. And you essentially just get the best of both worlds. So it's fast and it's accurate. And to tie this back to drug discovery, you can use this to predict likely sites of CYP450 metabolism. So often P450s go for the weakest bonds. Predicting those with quantum chemistry might take hours, and now it takes seconds with neural network potentials. And you can do it all through Rowan. So I think this is a pretty exciting time for people who are interested in studying fundamental reactivity of organic molecules, just because it's gotten so much faster to do work than it was when I was a grad student. I want to spend a little time also looking at the concept of studying hydrogen bond donors and acceptors. So this is something that I think has come in and out of vogue several times, maybe in the past decades, just looking at the literature. But essentially, the idea is that just like we quantify acidity or bond strength or basicity on scales, like pKa or bond dissociation energy scales, we can also quantify how strong a hydrogen bond donor or acceptor are through a similar scale. And all this requires doing is taking sort of a donor and a model hydrogen bond acceptor. People have traditionally used 4-fluorophenol, and then you just measure the equilibrium constant in a solvent. People usually use carbon tetrachloride because this was done back in the day and it's very spectroscopically neat. But you can show that the values measured here correlate well with those in other solvents. So you are learning something fundamental and not just something that's only true in carbon tet. When you do this study for sort of a large library of several hundred hydrogen bond donors, you can get out these values on a scale. And so you can say, cyclohexene is a very, very weak hydrogen bond acceptor. A trimethylamine oxide is a very strong hydrogen bond acceptor. And most functional groups that you would see in a small molecule therapeutic are somewhere in between. So, nitriles aren't too good, alcohols are fine, carbonyls are a bit better, amines or pyridines are a bit better than that, and so on and so forth. And these turn out to be pretty useful things to know. So if you're considering a scaffold hop, for instance, and you have a key hydrogen bonding interaction, you might want to match the strength of the two scaffolds you're hopping between, or try to modulate that somehow. So if you want to strengthen an interaction, you might want to use a stronger hydrogen bond. Or if you want something to be desolvated more easily and pass through a membrane, you might want to choose weaker hydrogen bond acceptors. This has been done, people have developed computational protocols for doing this simply because the experimental determination is not very easy to do routinely. So Peter Kenny and coworkers published this lovely paper back in 2016. And what they essentially showed, it's a very clever piece of work, is that you can look at the places on the DFT predicted electrostatic potential surface where the potential is negative. And that essentially corresponds to lone pair regions. And if you just take the minimum potential in the lone pair region, and then you scale that to match experiment, you get something that's actually very, very predictive of how good a hydrogen bond acceptor it will be. So here, if we look on this molecule, we can see that there's this N lone pair, there's this N lone pair, and then there's this sort of cloud around the amide here, and you can see that these are actually where the molecule will accept hydrogen bonds. These are the free electron clouds. And then you can sort of look at the values in here and predict how strong they'll be. So a customer of ours asked us to take a look at this. And the issue that we found with this workflow was essentially just that it was really slow. So it required a lot of quantum chemical calculations. And so what we were able to do was to build a more modern sort of, I'd say, higher throughput version of this using neural network potentials, where we can do all of the conformer screening and optimization without actually having to touch a single quantum chemical calculation. And then all we have to do at the end is just run a single R2scan 3C electrostatic potential calculation and use that to build the electrostatic potential surface and get all the acceptor sites and strengths. And so now instead of taking like hours and hours, this takes a couple minutes at most. You know, for fragments, obviously this calculation, if you run it on a protein, will be very, very slow. But for typical small molecules, this is quite quick. And we get here a very, very good match, I'd say, to experimental data, as good as the other ones that are published. And even in the cases where we see outliers, there are cases where there is a lone pair, but it's experimentally not accessible. So something like triisopropylamine, where we're accurately reading out that there's an N lone pair, but it's not something that's very easy to find. And so we were able to go back through and sort of look in the literature and see that we can actually reproduce experimental shifts in hydrogen bond strength usefully. So we see as we sort of walk this nitrogen around from this compound to here and then to within the ring, we can see that this move makes the two nitrogen sites here, increases their hydrogen bond basicity, and this move decreases their hydrogen bond basicity. And what we see over here is exactly that, so that the log D goes down in this case, and it goes up in this case. And then the permeability goes down in this case, and it goes up in this case. So I think we put more case studies in our paper, but I think this is just a nice illustration of how understanding something pretty fundamental about your molecule, how strong of a hydrogen bond donor or acceptor will be, actually proves to be quite useful in a modification that, frankly, I would not quite know how to predict the effects of these just by looking at these structures very easily. And of course, this is all available via code and on the web, so you can actually see where the acceptor sites are and make reasoned intuitions about them. So this is, just for acetaminophen, this is where the hydrogen bond acceptor sites are. This is the strongest one, perhaps obviously. And then this is where the donor sites are. And here we're predicting that the phenol is a stronger hydrogen bond donor than the amide, which makes sense. One of the things that people have been using this for out in the world, so this is a LinkedIn post from one of our users who's a VP of MedChem, I believe, at NRG Therapeutics, is actually showing that the molecular hydrogen bond basicity can predict experimental efflux ratio values. And so molecules that are stronger hydrogen bond acceptors are much more prone to efflux. So I think that's it's nice because it's someone else showing that this works, it's not us, but I think that's a nice way to sort of illustrate, again, something that's pretty complicated to mitigate sometimes efflux and how a pretty simple chemical insight can help you design around this problem. Okay, so for the last portion of the talk, I just want to focus in on sort of some of these, very important and trickier issues, which we're kind of segueing into, I guess, with the talk about efflux. And so this is just thinking about ADME and toxicity, right? So how do we deal with these? And how do we start to model these that start to matter more as we actually get to the realm of stronger binders? And so we at Rowan have been working to sort of build out a toolkit that can help people in this area with tools that we think are generally applicable. So I think one of the things that's worth saying about predicting ADME or toxicity is that it's often very series dependent. And so if you spend a lot of time working in a given chemical series or a given regime, you will probably build up an internal data set that will let you build your own predictive models. And they will probably, for some amount of data, always be better than something you can get from a team like us, right? Because we don't have your data. Nobody out there but you has this data. You are probably the best person in the world at some threshold of data to predict how your compounds will behave. And we don't, I think us and everyone else should be careful not to presume to talk down to people who have the data, because at the end of the day, that's what really matters. All that being said, before you get that much data or in conjunction with that data, there's still useful things that we can do to try to help. So we've recently started working a bit on predicting membrane permeability. We open sourced a tool called PyPerm, which is essentially a re-implementation of a a tool called PermMM, which is just a simple physics-based permeability predictor tool. So it builds a simple physical model of the free energy of membrane transport. You can see a membrane energy profile here, where there's getting over the membrane requires a certain amount of energy. And then it sort of uses these to predict intrinsic permeability values for different sorts of membranes you might find, like Keiko-2, the artificial Pampa, black lipid membrane, so on and so forth. This we think is useful. Other people have found it to be useful, and so we're excited to sort of just make that easy to use for folks. And we've also released a membrane permeability method that is based on just published data from AstraZeneca. They trained a nice model, and we have made it easy to use through Rowan. Moving sort of to the specific task of blood-brain barrier permeability, which is totally unimportant, except if you're designing CNS-targeted therapeutics, in which case it becomes very important. We read this paper by Schrödinger, actually, by Morgan Lorenz and coworkers, and found that they had a really, really clever way to predict the unbound brain-to-plasma ratio, which is essentially one of the ways to quantify blood-brain barrier penetrance, and I think probably among the most useful. And the way that this works for them is that they do conformer searches, they run a bunch of DFT calculations, and then they sort of take the difference between the vacuum and the water energy and use this as what they call ESOL, the energy of solvation. And what they find is that the energy of solvation is actually a very, very good predictor of whether or not something will get through the blood-brain barrier. Essentially, just for the simple reason that if it's more aggressively solvated, desolvating it to get through the membrane is much, much harder. And so as a part of this, we couldn't do this. Part of this requires a lot of pKa data here to actually state correct to get to a neutral state. And Schrödinger often uses experimental data to do this, or other sort of pKa calculations. And as part of our macroscopic pKa publication, we realized that by combining that workflow with some of our neural network potentials and low-cost solvent models that we've been working with, we could actually build a high-throughput way to do this that was, I think, on the order of 100 times faster, I estimate. And so one of the things that we've been able to do at Rowan is actually just to compute the solvation energy for people who run macroscopic pKa calculations, then quickly use that sort of to do a logistic regression model and binarize that to predict whether or not something will be able to get through the blood-brain barrier. And we can benchmark this on historic data sets and show that it's not perfect, right? Again, your own data for your compounds will be better if you have enough of it, but that it's quite useful, that it gets the right result a good amount of the time, and I think makes a ton of sense as a triage filter for the right stage in the project. Solubility is something that matters a lot. It's sort of irritatingly difficult to predict in many ways, and so we have a couple ways to do solubility through Rowan. So you can predict organic solubility using Jackson-Burns FastSolve with these nice per solvent calibrated uncertainty values. So if I look at sort of this high throughput hit, this is just data I pulled from something I was working on, we can see that this molecule is predicted to be very, very sparingly soluble in hexane, very nicely soluble in THF and ethanol, and somewhere in between for the likes of toluene and ethyl acetate. So this is nice if you're planning to do a screen or if you're trying to assess something synthetic. We've just published a good amount of work that we've done on aqueous solubility prediction. It's hard. We tried to do a good amount of benchmarking. So this paper is out for you to read. We benchmark a bunch of different architectures. And as a part of that work, this is actually, I showed a screenshot of this earlier, but we've shown that we can predict pH-dependent solubility quite nicely with some of these machine learning models in combination with pKa data. And then we sort of have, we've adapted some pre-trained models from Kyle Swanson just to predict a bunch of other ADMET properties. And these are like global ADME-tox models. They're not a substitute for experimental measurements, but I think they're quite useful in the right circumstances. And so you can just pass a compound through. In a couple seconds, you'll get back predictions for all of these sorts of things and say, this molecule is throwing a flag for drug-induced liver injury. I don't think this result means it will cause drug-induced liver injury. That doesn't necessarily mean that, but it means that the model thinks it has a higher chance of that than a regular compound. And so it's at least worth digging into, thinking critically about, and maybe using as a first pass flag for things like this. Sort of the last scientific feature I want to talk about today is free energy perturbation, the gold standard for computational binding affinity. And this is something which I just moved to the end because it's not out yet, but I'm so excited I just want to be able to talk about it. We've been working with a really fantastic scientific team to build out a low-cost free energy perturbation methodology. For the unfamiliar, essentially, this is just a way to evaluate the changes in binding affinity along a series. So you create this like graph where you have a bunch of analogs and you group similar analogs together, and then you run alchemical free energy calculations to predict the difference in binding affinity between these two compounds when they're bound to the protein. And from this, you can sort of take out a list of all of the ligands and what their predicted delta G will be. This is traditionally really expensive, so you'll see numbers like 10 GPU hours per compound floating around. So this means it costs a lot of real-world money to run just in computer time. And we have been able to get this down to about 10 to 20 minutes per ligand, I'd say in most cases, sometimes up to about 25. So we're able to benchmark against state-of-the-art programs and show that we get, comparable errors. This is a very easy system. Take two, there's many harder systems which have higher errors, just because the calculations are not capturing various properties of the system or the force fields are insufficient. But the thing that we're really excited about is just to be able to bring this down so that the cost is low enough for routine usage for people. And so we'll be releasing this later this year. And I think this is the thing that our company is by far the most excited about right now and what I'll be working on after this presentation is done. That sort of brings me to the end. I think the picture I've hoped to paint is just that there's a lot of tools in Rowan that we are not claiming by any stretch to be the smartest people on Earth. There are many people smarter than us. There are people who are experts in specific fields. There are people in the audience with much more knowledge than me on aspects of this, I'm sure. I know there's some permeability experts there. What we've tried to do is just bring together what we find to be all of the tools that we think the modern drug hunter should have at their fingertips and make them easy to use, interoperable, so you can just focus on the science and not on building out the software package. So it's just coming back to the toolkit idea. Sort of towards that same thing, in service of that same goal, there's also experimental, I'd say, integration that we have between Rowan and large language models using the model context protocol. So if you want to connect Rowan to Claude or something else and try to ask it to run calculations for you, this is being developed actually by another company, by Kat Yenko from BioArena. She's a fantastic engineer. And so this is, I think, we're trying to figure out still the best way to bring sort of integrate computational tools with AI. But I think there's a lot that will be exciting there, and I hope to have more to share later this year. We can work with organizations. I don't need to get this, but it is encrypted and all this stuff, and you can control that. But what I want to just end with is like, if this is something that you guys want to try, or if this is useful, just check out Jonathan's tutorial tomorrow. It's totally free to try Rowan. We're not here trying to sell licenses or anything. If you are working in a company and you want to try to collaborate with us, let us know. And then we also work with scientific teams in academia and industry on open source projects. So we've open sourced a variety of things. I've shown some, but there's other open source tools on our repo like conformer deduplication, symmetry correction, other pieces of infrastructure that we just think should be open. We're working on an open conformer search engine. And if there's something that you think would be useful to Rowan, that is a tool that you'd be willing to work with us on open sourcing, let us know, and we're totally happy to talk. So yeah, that brings me to the end of my prepared remarks with hopefully enough time for some Q&A or yeah, just to give you guys that time back for your conference. Thanks so much for listening.
Thank you. Can you see me?
Yeah, I can hear you.
Yes.
So can you hear me? Okay, nice. I would like to ask you about the potentials from machine
learning and how do they act with solvents? Because usually they're trained in vacuum and you have something like UMS or others. And sometimes they also work as corrections. For example, there's a new one that works as a correction, delta learning to GFN2. So if you have some experience or Rowan provides some potentials that would work in solvent, because, for example, proteins are there in a solvent and you would like to know their conformational energy and stuff like that. So how is it handled? Thank you for the answer.
Yeah, it's a fantastic question. I actually added this bullet point here, but forgot to talk about it. We could spend slides on this, but you're right that the core issue is that these potentials are almost always trained just on gas phase data. And so then you get gas phase energy for the thing you care about, which is not usually super relevant for things in water. This is not the solution that we've been using at Rowan is not what will be the best solution in the world. People will come up with better solutions for this. I talk to people who are working on this, and I can't wait for them to be done. What we do is we run semi-empirical calculations behind the scenes, and then we essentially just do this as a multi-layer correction scheme. So we just take the correction from energy and forces from GFN2 XTB with ALPB, the Poisson-Boltzmann model, or with CPCMX, and then we apply that correction on top of the NNP. And so that lets us do things like actually model charged reactivity. So if you want to model like a cysteine anion attacking a Michael acceptor in a Thiomichael reaction, you can do that once you do this correction, and that reaction doesn't exist without that reaction just doesn't work in the gas phase. You get like an electron transfer instead. So the semi-empirical solvent correction works for small molecule reactivity. It totally does not work for proteins because you can't run GFN2-XTB on a protein and expect it to behave properly. The issue is that most of the neural network potentials don't work that well for proteins either, so it's not that big of an issue yet. But I think what we're going to need ultimately is like a separate machine learning solvent correction model. And Bin Zhang has been working on this at MIT. I know Serena Riniker has been working on this a little bit at ETH. So I think we'll get something that will work for this in the next year. There's just nothing that I'm aware of yet.
Hi, great platform what you have. And I would like to ask whether apart for science, whether there is some chance for teaching, whether you had in this direction. Thanks.
Yeah, it's a great question. We work with, people use Rowan for teaching a lot. I can think of like 3 big classes right now that are using Rowan for lab assignments. And we've done more. We've done grad classes, we've done undergrad classes. We provide some example materials. So there's a section of our documentation that has like example laboratories. So if you wanted to teach a conformational analysis homework, like if you wanted to give electron conformational analysis and then give homework for the students, how would they do it in Rowan? That would be fast, simple, easy enough to do. So we have example materials out there, but we generally leave that to the TAs and professors to ultimately figure out exactly how to integrate that with the course. So we haven't taught our own classes yet, just because we're pretty busy, frankly. But we're happy when people do that with Rowan. I think that's something we as a company care about making this accessible, because I remember how much it was painful to learn computational chemistry at the terminal. And so, I think it's something that is a feature of this for sure.
I'm gonna pass microphone to myself. I have actually a question about the membrane permeability. I've seen you using Piper, which is still not like feature complete implementation of Perma. And you've said in the newsletter that there are some deviations from the original photochemical growth, which is used by Perma. So whether this is going to be addressed. And then a second question, whether you are aiming to add more modules such as GNN and MTL.
Good question. We do have GNN MTL in Rowan. It's not on the slide, but it's there. When you submit a calculation, you could just choose from a dropdown how you want to do it. I just was trying, I thought if I had 300 slides, you guys might lose your attention, which I thought was fair. In terms of PyPerm, in our hands, we've done benchmarking back and forth with the Fortran perm/mm. So we took, for the people who don't know the context, we essentially took a program that was in Fortran and like transpiled it into Python. We benchmarked back and forth. We ported the small molecule logic, but we didn't port the peptide specific logic. So they have a special handling for like oligopeptides that we haven't moved over. We may do that in the future, or if someone else wants to, it's open source. You're welcome to open a PR. For us, we don't really work with companies that are doing peptide modeling much. Or if they do, they're doing like peptidomimetics, so the canonical alanine residues, like the residues are all different anyway. So the logic from perm/mm, we just didn't bother dealing with just because it wasn't relevant to us. So I think it's not feature complete in that we haven't done all the things, but I think in the areas where we have ported the logic, it is correct as far as we can tell, and we've tried to run as much sets through it as we can. So it's not a short-term fix. Maybe at some point in the next year we'll get around to doing the rest of it. And I think we're always looking for new permeability prediction methods. So if there's good stuff out there that we're missing, especially that's like theoretically complementary to GNN, MTL, or PyPerm, I think having more different methods just gives scientists more tools to like draw consensus from. So that's something we're definitely interested in.
So thank you for your answer. Maybe just a question, one final one, whether like something like Cosmoperm is planning to be, or an alternative of Cosmoperm is planned to be implemented in the future.
It's not on the short-term plans, but let me dig into it and see. I thought with something like Cosmoperm, the license just wouldn't work for us, because we don't have a Cosmo RS license yet. But let me just dig into it and see if I think it can work.
Thanks a lot.
I'm writing it down.
Hopefully it will.
Where
should
should
I
I
Sorry, there is quite a long lot of feedback here in the aula. So my question would be, you've started with batch docking, but the batch docking is not available. So when it will be available to you?
Yeah, great question. I need to figure that out. It's available right now. We've rolled it out just to organizations because it's expensive and we're trying to make sure it works well. So I think this will be more generally available at some point, but we're trying to make sure that it works well enough there first. So we often roll things out in stages at Rowan where they'll come just to one person or a couple people first. Then we'll open them up to a smaller subset of people, and then we'll move towards general access for them. Simply just because I don't like it helps us to iterate and test and make sure that things are solving the scientific problem we need. And it's easier to learn from one person you're working with than from 8,000 people when it breaks for all of them at the same time. So that's a non-answer, but I hope soon. I can't hear anything you guys are saying right now. I don't know if that's
Yeah. Thank you.
Okay. Yeah, thank you guys so much. Thanks for the invitation, and I hope the conference is fantastic. Hope to be there in person sometime.
Yeah. Hopefully next time. See you.
So, after a Zoom session, we will have another session, and this time it's a poster flash talks. So I hope that we have one additional flash talk about the workshop, which will be on Wednesday, I think. And then there are three poster talks. So please do come if you are the ones who are here. And You will have your feed. So I hope.
Hi, I hope you can hear me. If not, I'll just talk louder. Yeah, short introduction. We just want to hope and motivate you to come to our workshop we have on Wednesday about our simulation and analysis platform for MD simulations called OpenMMDL. Just because we were asked to give a short flash talk here, just to round it up, what we will be handling, we'll be showing you how to set up your MDs with OpenMMDL, not really how to run them because that might take too long for the workshop, and then we'll show you what the analysis done in the end with the example I prepared. I'll upload the link or give the link in the Discord later this day so you can already have a look if you want to. And then during the workshop, Valerie will also give a short information about MD analysis, what you can do with it, and also ProLift, and maybe he'll talk shortly about that.
Correct. So it's basically the case that on Wednesday it's 2 parallel MD simulation workshops, and they are both connected with MD analysis and ProLift. And what I'm going to do is basically that I will be present in both of the workshops to kind of first showcase what is new in MD analysis, with this case being the parallelization, because again, there are many analysis tools in MD analysis, and most of them were serial. And now basically it's the case that MD analysis tries to one by one kind of make them parallel, either using multiprocessing and Dusk. And what we're going to do is basically I'm going to prepare a notebook that you're going to see and use to basically see that the performance is now faster. The second thing is the ProLift, which is again now both in OpenMDL and SimMD. And there is the case that basically ProLift didn't have water bridge interactions, so it had different interaction types that you could calculate, except for the water bridge interactions. And basically what I'm going to do is kind of then showcase how they work now, because again, this was an addition that was implemented only last week.
Thank you. So that was mine example of how the poster presentation should look like. So let's skip to someone who actually has a poster presentation. And that's Alexandra.
Workshop as well.
And workshop as well. And that's Alexandra Ivanova from Pavel Poliszczuk team.
Hello, everyone. Yeah, today I would like to talk about our tool for high-throughput molecular dynamics simulation, which we implemented, StreamMD. And the idea to implement this tool was very simple. We wanted to run not 10, not 100, but 1,000 and more thousands of simulations very easily and very efficiently. And to address this, we did. And to address this, we implemented such features, for example, as that we can run, for example, on distributed servers or on GPU node servers, which can give you very good boost with the efficiency. As well, you can easily continue your interrupted or unfinished simulation. Sorry. Which is also a very important feature because it's time-consuming calculations. And last but not least, it's not just return your trajectories for these thousands of systems, but also some automatic simple analysis such as RMSD, RMSF, etc. It returns you some CSV tables as well as some plots, so you can take a look what is going on in your simulations actually. As well, it supports some more advanced analysis such as ProLift for protein ligand interaction fingerprints analysis, or GMX MBBSA, another great tool which we wrote a wrapper around for the binding free energy calculations. As well, you can run it for thousands of your system very quickly and very easily. And how you can do this? So there's example. You can run your 1,000 simulation just by one command. So run MD. The user should provide the protein in PDB format. If necessary, SDF file with your ligands. The user supposed to set up the MD time of simulation in nanoseconds, and the replicas, because reproducibility is very important for the molecular dynamics simulation. So in this case, it's 10 replicas. And yes, so that's all. Now you set up your 1,000 in 10 replicas for them, and you just wait for your results, which, as I told, gives you very nice plots with aggregated results as well as tables, aggregated tables, which you can analyze further. So thank you for your attention, and you're all welcome to come to my workshop, which will be also on Wednesday in parallel with OpenMMDL tool in room 3.002. So also there is a poster upstairs. You can also take a look if you don't have time for workshop. So thank you very much.
Thank you. And we'll switch for Guzel, Minibio. And speak more closely to the microphone.
Oh, okay, thank you. Good afternoon for everyone. Cycle-independent kinase 16 is involved in cell cycle regulation and overexpressed in several cancers such as lung, breast, liver. To identify potential inhibitors of CDK16, we utilized Yanamine Real database, which contains 10 billion, approximately 10 billion enumerated synthetically accessible compounds. Given the impracticality of exhaustive screening across the vast library, we implemented two-step procedure to enhance similarity search efficiently. Initially, we generated compounds by CREM-doc approach, a fragment-based generative method guided by molecular docking. Then molecules were chosen based on their superior docking scores and ability to interact with hinge region. We use these molecules as query structures for rapid similarity search across the entire database. Then the best molecules were resubjected to consensus docking against CDK16. In our project, we investigate the influence of fragment source, the synthetic accessibility of generated templates, and descriptor type on efficiency of retrieving promising compounds from Yanamine database. Our findings indicates that the native queries, the compounds which generated from Yanamine fragments during the first stage yielded molecules with improved docking scores in the second stage. Also, furthermore, atom pairs fingerprints leads us to superior performance in case of docking scores. That's why we used molecules which found by atom pairs queries for the future purchase and future experimental validation. Thank you for your attention.
Thank you. And we'll switch for Kaveri Prasad.
Hi everyone. This is my research project. I've been developing predictive models for photoswitches. So photoswitches are molecules that can change from one form to another when irradiated with light. We have been working on EZ photoswitches, among which azobenzene or azo photoswitches are the most common. So azo photoswitches, as you can see, exist in two states: a trans state that is a stable one and a cis state that is a metastable one. So when irradiated with light of a specific wavelength, these forms are interchangeable. Because of these properties, they are used immensely in materials and medicines, especially in case of drug discovery. These are highly important because they provide spatiotemporal regulation and non-invasive activation. But discovery of these photo switches is a tedious process because we need to tune their photophysical properties to use them for specific applications. To date, structural features are the only ones that dictate these photophysical properties, which can only be understood by synthesis or by quantum chemical calculations. But these are time-consuming and expensive. This is where machine learning comes to the picture. But most of the models that use these uses DFT calculation at least once in their development or data curation. But we have developed a machine learning model that can predict these properties using structural features. So the input data only consists of smiles, the property value, and the solvents because solitochromic effects are very important in determining these photophysical properties. So as a part of our workflow, we collected data from literature. We also performed some experiments to get the properties, converted these into some descriptors for both structures and solvents, developed models, validated these models using some novel ASO derivatives, and compared their experimental and predictive values. So I have explained the benchmark study, the selection of descriptors, results, everything in my poster. So if you'd like to discuss, please come and visit my poster. It's poster number 13. Thank you.
Thank you. And that's it. We don't have any further flash talks for the posters. So let me finish this session today by thanking you that you survived all of those lectures, starting with mine, of course. And now what we will be having is that since we had a poster flash talks, now you can enjoy the poster themselves. They are in the third floor, so if you leave the room from the upper part, you will directly walk towards the posters and you can enjoy them. And they will be there for the rest of the conference. So you can enjoy them for a whole week. Also, I would like to thank all of the speakers of today, including the poster flash talks, and I've 3D printed a small gift for every lecturer and speaker, so you can come and collect your 9ADD statue. And I think that's all from me today. So have a nice evening. Enjoy the posters, enjoy the stay in Olomouc, and see you tomorrow.